Very good morning to all of you. First and foremost, I would like to congratulate each and everyone for celebrating the essence of IAPEN Foundation Day today, India Day today. And I would also like to thank Professor Dash, President IAPEN, for inviting me today as a chairperson. And it would also be very pertinent to mention about the words of wisdom shared by Dr. Vijay Lakshmi. Madam, you are very right that it is the right time that we have to recognize the importance of nutritional care process terminology. Everybody is aware about nutrition care process, but terminology is important. It is being adopted by many countries internationally, and now it is time. It is a time for India to adopt. Then only we can move ahead as per international standards. And it is also time to recognize food is medicine. And for this, we have a very best speaker, Dr. Nirja Hazela. She is Head Science and Regulatory Affairs, Yakul Danun India Limited. Dr. Nirja Hazela has done her PhD in biotechnology. She is currently the Head of Science of Regulatory Affairs and Key Quality Assurance PR at Yakul Danun India Private Limited. She has over 18 years of experience and has worked in organizations like Ranbaxy Biotech and Limited. She is involved in the development, planning, and implementation of scientific programs, including clinical trials for the company. She handles all the regulatory issues and ensures compliances related to the category and individual products. She works closely with healthcare professionals and scientists to advance the science, promote evidence-based clinical use, and improve consumer understanding of probiotics.
Thank you, Dr. Tanveer.
Over to Dr. Mirza Hadela.
Thank you so much. Thank you very, very much. And it's such a pleasure because I'm seeing so many known faces after a very, very long time, literally after COVID. And so to begin, dignitaries on the dais, of the dais, uh, fellow colleagues, friends, and students. At the very outset, I must thank Dr. Dash, Mrs. Jitana, Dr. Vijay Lakshmi, Shilpa, and of course everyone who is associated with IAPEN for organizing this very timely symposium on probiotics and prebiotics. So we are actually going back some 2,500 years back, and even in the Ayurvedic times, we all believed that all the diseases begin in the gut. Hippocrates said this about that, a bad digestion is the root of all evil. But having said that, the gut has been one of the most neglected organs of the human body. But again, in the last decade or so, the spotlight is again on the gut. And like we heard from Dr. Vijay Lakshmi, it's all to do with the gut microbiota. So I'm going to be talking on a very different topic, the gut-brain axis, and what does it really mean. Contents, I'll talk about the gut microbiota. I'll go back to basics. Gut dysbiosis, is it linked to stress, anxiety, depression? What is this so-called gut-brain connection? Probiotics, can they really help? And a take-home. So the last decade has been fascinating. And we have actually entered into a new era of science where we've heard that we harbor trillions of microbes. And these microbes are present in almost every part of the human body, like Dr. Dash said, lungs, but also the intrauterine environment, which was initially considered to be absolutely sterile. Now, if you look at the number of microbes, they are, at one time, we believed 10 times the number of human cells, but today we know they are exactly the number of human cells. Now let's look at the genetic composition. The human cells have 23,000 genes, and the microbes have a staggering 3 million genes. So it brings us back to the question, who is the host? Whether we are hosting the microbes or is it the other way around? Now, the largest number, like we also heard in the previous talk, is of microbes are found in the gut, the intestine. And as you can see here, the microbes are very unevenly distributed. And this is because of differences in pH, in transit time, redox potential, and substrate availability. So the stomach harbors the lowest number of microbes because of the secretion of harsh gastric acids. And as you go down the duodenum, the jejunum, and the colon, you have almost 10 to the power 12 microbes per gram of the colonic content. Now, the next question is, what are these microbes doing in the intestine? So, they have very, very important protective, structural, and metabolic functions. So, these microbes, believe it or not, are your first line of defense against pathogens. They inhibit the proliferation of pathogens by competing for nutrients, by competing for receptors, and also producing what is known as antimicrobial compounds like bacteriocins. Now, again, a very interesting fact is that about 70% of the body's immunity or the gut associated lymphoid tissue is located in the intestine. And the reason for this is simple, because the gut is your first point of contact with the external environment. And so therefore, it has to have its own defense mechanism to recognize what is important, what is not, and act accordingly. So these microorganisms play a very, very important role in building your immunity, in the proper growth and development of your immune system. They help in inducing the secretion of what is known as secretory IgA. Now, secretory IgA is a very, very important antibody which is found in all the mucosal linings. So your sweat, your tear, your saliva, your GI tract, all has this secretory IgA. And one very important fact is that we all know that our intestinal epithelium is just one cell thick. So it is vulnerable to damage. It can be distorted at any point of time. So these microbes are playing a very, very important role in producing what is known as tight junction proteins. And these tight junction proteins, they hold the cells together and ensure intestinal barrier integrity. Now, we are all nutritionists here. One very important function of these microbes is they help in the digestion of foods that cannot be digested by the human body. So if you look at your non-digestible fibers, your starch, and your oligosaccharides, it is these microbes which have the enzymes to digest these foods and convert them into what is known as short-chain fatty acids. And these short-chain fatty acids are now a very important source of energy for your common cell bacteria. And the presence of these short-chain fatty acids reduces gut pH, it improves gut motility, and also inhibits the growth of pathogens. Now, as you can see here, these short-chain fatty acids makes the environment of the gut so conducive for better absorption of minerals like magnesium, calcium, and iron. And incidentally, many of these microbes also have the ability to produce what are known as vitamins like vitamin B, K, and folate. And a reduction or a vitamin deficiencies have actually been linked to a reduction in some of these common cells. I danced a lot last night, so my throat. So now this balance is very, very important. So we have the good, the bad, and the ugly. So the number of good bacteria have to be in much higher numbers as compared to the bad. But what happens in today's day and age, in with this kind of a lifestyle, there is a total dysbiosis or an imbalance in the gut microflora. And this is because of conditions like poor nutrition, poor bad eating habits, infection, and like Dr. Vijay Lakshmi said, antibiotics. So antibiotics are chemicals cannot recognize between the good and the bad bacteria. So they kill both the bad as well as the good. Alcohol, stress, and aging. So now, interestingly, if you see this imbalance or so-called dysbiosis is linked to diseases. Now, this is a chicken and egg situation. We really do not know what came first, whether the imbalance in the gut microbiota resulted in disease or whether the disease is linked to this imbalance in the gut microbiota. But the association is very, very clear, and almost 25 different disorders are linked to this imbalance in the intestinal flora. So, metabolic disorder. Now, how do you connect a metabolic syndrome with the gut? But just look at the connection. When you take a diet which is very high in fat, so what happens is it allows for the growth of these harmful bacteria. And these are gram-negative bacteria which have an endotoxin which is known as lipopolysaccharide. Now, when there is the gut dysbiosis, also your intestinal permeability is affected. So there is a disruption in the gut barrier, and it allows for the entrance of all these endotoxins into your circulatory system. And when this enters into the circulatory system, it causes tissue inflammation, insulin resistance, and this is linked to metabolic disorder. So today, almost every disease condition, look at liver, look at heart, look at lung, look at skin, is all linked to an imbalance in this gut microbiota. Now, the gut-brain axis. So again, the book, the concept has opened up on the gut-brain axis. So it is believed, and this was known forever because the concept was introduced way back in 1880s, and it was introduced by William James and Carl Lange. And it was there, but again, in the last decade, there's been intensive research because of the advent of these sequencing technologies, metagenomics, which has helped us understand what really resides inside. So it is believed that there is a bidirectional communication between the gut and the brain through the vagus nerve. So signals from the gut, they actually can modulate your motor function, sensory functions, and motility of the gut. And on the other hand, signals from these gut microbiota can actually influence emotions like anxiety, pain, and stress. So the proposed hypothesis is this, that you have intraluminal bacterial cells, you have metabolites, you have dietary components. So what we have is we have these enteroendocrine cells which can actually sense these different kinds of substances. And the immune cells which are found in the gut also can sense these different types of substances. And what happens is they convey their signals through the sensory nerve or through the bloodstream directly to the brain. And because of the presence of LPS, fatty acids, sugars, XYZ, this vagus nerve controls that information to the brain. So this is the proposed mechanism of how it works. And mucosal barrier. So that integrity of the intestinal wall is very, very important because it's only one cell thick. So any damage to the intestinal wall will ensure all these components, so-called harmful components, come directly into the circulatory system and they cause this kind of a damage. Now I'm shifting gears. I'm going to talk about one disorder which we've heard about a lot, especially in the COVID times. This is depression, also known as major depressive disorder. It's a common mental disorder and it is characterized by symptoms such as depressed mood, fatigue, anxiety, irritability, insomnia, also sometimes suicidal thoughts. So currently it affects about 280 million people, and there, of course, the cause or the pathogenesis of depression is multifactorial. We all know about the monoamine hypothesis, which has also resulted in the development of antidepressants. But given the gut-brain connection, there is a possibility that there could be a link between the gut and so-called depression. Now, the first proof of concept came from fecal microbiota transplant studies. So for students who are here, fecal microbiota is nothing but transplanting the gut microbiota from a donor to a recipient. So you can actually transmit diseases. So this study used germ-free mice, and germ-free mice are mice which have absolutely no microbes. So you had two mice, you had one depressed patient, you had one healthy control, and the gut microbiota was transplanted into the germ-free mice. And what happened was, and the microbiota from the healthy control was also transplanted into the germ-free mice. And what was seen was that the outcome was very different. The composition of the microbiota was significantly different in the germ-free mice from the depressed patient as compared to the healthy control. And when these mice were forced to do the swimming test or the tail suspension test, which are actually stressors, you saw that the germ-free mice with the depressed microbiota had much more depression-like behavior as compared to the healthy control. So probably the gut microbiota is involved. Now, like I said, in the last 10 years, there have been lots and lots of studies. And what this is just a systematic review of studies which have been done on the gut-brain connection. And these were 17 studies. They included 738 depressed patients. I'm going to call them MDD patients and healthy controls. And as you can see here, in 8 of the 17 studies, there was a decrease in Faecalibacterium prausnitzii. Now, Faecalibacterium is a butyrate producer. And I go back to short chain fatty acids, where the butyrate production helps in maintaining the intestinal barrier. It reduces inflammation and also helps in neurogenesis, or the development of the neurons. So 8 of these studies showed a reduction in the Faecalibacterium. This is one organism. And again, this is a study which was conducted by Aizawa et al. And they actually just checked the gut microbiota of MDD patients and healthy controls. And what was observed, there were 43 MDD patients and 57 healthy controls. And there was a decline in bifidobacteria and lactobacillus in the MDD patients as compared to the healthy controls. So while we cannot conclude that the gut microbiota, a particular organism, is linked to depression, but a decrease in some of the microbes, but the gut microbiota is kind of altered in patients who have depression. Now let's look at this hypothesis, which talks about the gut dysbiosis and its link to reduced neurogenesis, abnormal stress response, and neuroinflammation. So this is a very complicated slide, but let me take you through this a little slowly. So what happens is, when you are stressed, your HPA, the hypothalamus, pituitary, and adrenal axis, gets activated. And what happens is, this activated HPA axis stimulates the hypothalamus to secrete what is known as CRH. And CRH then stimulates the pituitary gland to secrete what is known as ACTH. And this ACTH is then released into the circulatory system. ACTH produces cortisol, and this cortisol is produced by the adrenal cortex. Now, what happens is, this cortisol has its own feedback mechanism. So what happens is, it can inhibit the activity of the HPA axis, and it can also inhibit the activity of CRH, and therefore reduce the secretion of cortisol in the circulatory system. But for MDD patients, there is a negative feedback. So this negative feedback is impaired. So the cortisol is constantly there in the circulatory system. The brain is constantly being exposed to the cortisol, and as a result, there is inflammation, there is decreased neurogenesis, and there is decreased BDNF production. And all of this is again linked to the pathogenesis of MDD. Now let us look at how the bacteria can respond to this particular response. So this was again germ-free mice, one, and specific pathogen-free mice was the second group. And what was done was, these germ-free mice and the pathogen-free mice, they had bacteria but no pathogens, no harmful bacteria. So when they were exposed to stressors, and the stress markers were checked just before exposure to stress and after exposure to stress, look at this graph. It tells you that the specific pathogen-free mice which had the microbes, they had much lower levels of the stress marker as compared to the germ-free mice. So this again tells us that possibly the gut microbiota is involved in this pathogenesis of MDD. So these are the initial studies, and therefore there is an interest in the gut microbiota, in FMT, in probiotics, prebiotics, to just modulate the gut microbiota favorably and thereby reduce the symptoms of depression. So FMT is fecal microbiota transplants, and what has been seen that in IBS patients, when you basically did a fecal microbiota transplant and transplanted the microbiota from a healthy donor to an IBS patient, the symptoms improved. And probiotics, because of their ability to modulate the composition of the gut microbiota, increase the beneficial bacteria, decrease the harmful bacteria and the pathogens and the toxins, they could be useful. But then let's ask ourselves a question: what really are probiotics? And there has been a lot of confusion around this term. So the word probiotic means for life. They are scientifically tested bacteria which reach your intestine alive in very large numbers to give you a scientifically proven health benefit. So they're very different from your traditional foods which actually contain lactic acid bacteria but are not scientifically proven. And why I'm talking about that is because all the lactic acid bacteria which are found in these foods are killed in the stomach, in the gastric juices of the stomach. But probiotics are tested in the laboratory to be able to transit the GI tract and reach the intestine alive in very large numbers. Now, this brings us to the question, but is there enough clinical data for the use of probiotics in depression? And look at this. These are, again, a systematic review. And the whole concept of psychobiotics has been introduced here by Professor Cryan. He's from the University College in Cork in Ireland. And these are about some 34 studies where they have introduced the concept of psychobiotics, and this is for those live organisms which help patients who have psychiatric illness. And as you can see here, it is all going towards a benefit. So there are studies which have indicated that probiotics could be useful in treating your depression, not as primary treatment at all, but just as conjunct to other treatment. And but here you must remember that probiotic benefits are strain specific. So every probiotic organism is not going to do the same thing. It has to be scientifically validated. So I have seen products in the market which say probiotic curd or probiotic lassi or probiotic XYZ, but the strain is not mentioned. So you really do not know what is happening to you after consumption of that product. So it's very, very important to mention the complete name of bacteria, the number of bacteria which gives you that health benefit, and the scientifically proven health benefits. Now let me take you through some studies. So this is a study which is a double-blind placebo-controlled trial. It was conducted on MDD patients, 40 patients, intervention of 8 weeks. And this was a probiotic capsule which was given for 8 weeks. And what was observed was there was an improvement in depressive symptoms and insulin resistance markers in the group that got the probiotic. Yet another randomized, double-blind, placebo-controlled, randomized control trial, a 3-arm parallel design study, and again on MDD patients who were already on treatment with antidepressants. But when they were given the symbiotic, now this is a symbiotic preparation, that is probiotics and prebiotics, and they were given that there was an improvement in depressive symptoms, and there was also a reduced kynurenine tryptophan ratio. So we all know tryptophan is that one component which substance which basically goes the serotonin route or goes the kynurenine route. So if you have inflammatory cytokines, it goes the kynurenine route, it gets converted to quinolic acid which has neurotoxic effects. So there was also reduced ratio of these two metabolites. So clearly indicating that possibly they could help. Now let us look at this study. This is a before and after open trial. It was conducted on 15 patients with MDD and 3 patients with bipolar disorder. And the LCS fermented milk drink, which is commonly known as Yakult, 80 billion of those bacteria were given for a period of 12 weeks. And as you can see here, there was a significant decline in the stress markers and in improvement in symptoms of depression. And when they try to understand what was the gut microbiota composition, clearly what they said, they had 2 groups. They had responders and they had non-responders, and the responders had higher levels of bifidobacteria as compared to the non-responders. So bifidobacteria plays a very, very important role in, allowing for the probiotic to actually act. Now, this is the proposed pathway that we see. So when we have chronic stress, the stress response results in an elevated sympathetic activity, hypersecretion of cortisol. Cortisol is a stress marker. You have GI problems, you have immune suppression, you have sleep disturbance. And when you take a probiotic, this is just by way of example, I'm talking about LCS, but it normally normalizes or improves the balance of the intestinal flora, results in immune modulation, and thereby it can basically help your vagus nerve to send the right signals to the brain and thereby reduce depression and anxiety. So now coming to the last part of my presentation, when we are talking about health, that one health approach, we are talking about longevity, we are talking about good health, and we're not just talking about the absence of disease. So what is possible is that these probiotics, they are preventive in nature. They are not used for treatment of any disease, but what they can basically do is ensure the homeostasis of the immune system, the nervous system, and endocrine system, and thereby improve overall health. So probiotics do not act on single sites. They are not drugs, but on the whole, they act on the entire body through the immune and the neuroendocrine system from multiple intestinal sites in a direct or an indirect way. And the take home here is that gut microbiota is today being recognized as one of the key regulators of gut brain function. It has led to the importance of microbiota gut brain axis, and the use of probiotics is identified now as a very promising way to prevent gut microbiota dysbiosis, restore not only physiological homeostasis but also psychological homeostasis. And human studies, including ones that we have done with Yakult, demonstrate benefit of probiotics to reduce stress-related physical symptoms and sleep disturbance. So thank you very much. I acknowledge Dr. Kazunori Matsuda. He's a scientist at the Yakult Central Institute. You can see this institute there, and he has been very generous in providing me most of the slides. So thank you very much. I'd be happy to take any questions.
Thank you very much, Dr. Nija, for the excellent presentation, and you really shared very deep knowledge regarding the rationale behind the importance of gut microbiota, in attaining well-being in humans and also in boosting immunity. And what is your take on other foods besides curd and yogurt? We say they have good number of probiotic value. What about other food products? What is your opinion regarding that?
So I think probiotics is not your mantra to just probiotics is not your mantra to good health or to good brain function. I think it has to be balanced with a good diet, with adequate physical activity.
I was reading the book, and then they were mentioning that the role of bananas and
because they have contained tryptophan,
Yeah,
so
yeah.
probably the happy hormone, the
and even
serotonin
pickle-
levels
and the
increase.
pickle we take in India.
Yes, yes,
Pickle,
yeah.
they also have a probiotic value.
They're
But we don't
fermented,
fermented
so
foods in India.
yes. So while they are fermented and they contain lactic acid bacteria, they're excellent for their nutritional value. But those bacteria get killed in the stomach, and therefore they do not reach the intestine alive, and therefore they are not scientifically validated. But of course, they are excellent for the
strain you would like to mention that will be of more significance.
Of course, Yakult, I'm not talking about, but we have BSL-3. It is a cocktail of 450 million bacteria. So it has lactobacillus, it has bifidobacterium, it also has streptococcus. So BSL-3 is there, enterogermin is there, but they're all medicines. So sorry about this. So they are all medicines. They are normally taken for a limited period of time, and then after that what happens is you stop the medicines. But when you're looking at foods, any strain which is scientifically validated and for that particular health benefit, whatever it may be, there are strains which are helping. For instance, there is Lactobacillus rhamnosus GG, which is very good for acute diarrhea, for allergies. LCS works in immune modulation and reduces stress cortisol levels. Then you have BB12, you have LA5. There are a lot of strains, but it's just important that they should be validated scientifically.
Because we have to promote food as medicine.
Yes, yes, yes.
Talk about prawn.
So exactly, see, these are all fermented foods. So see, because you have a definition for so because you have a definition for probiotics, these are bacteria which are scientifically tested to reach your intestine alive. So sauerkraut, kombucha, kefir, all of them, they are excellent for nutrition, but they do not have probiotic bacteria unless and until that sauerkraut has the product has a declaration that it contains this particular probiotic bacteria in so many numbers to give you this kind of a health benefit.
Do you think when we, telling our patients, clients coming to us, like there are products also there who are selling sauerkraut as a product also?
As a probiotic. So you have
better
to
to make it home or telling them to buy those bottles
which contain the probiotic bacteria, not the ones without the probiotic bacteria. So those products which contain a particular scientifically tested probiotic bacteria, the number of that probiotic bacteria, the scientifically proven health benefits, and now FSSAI has made it mandatory. You actually have to write on a probiotic food, the name of the bacteria, the number of bacteria, scientifically proven health benefits, all of that has to be mentioned on the product. So if it is scientifically proven, absolutely.
So homemade ones do not give that
Do
much
not give you the probiotic benefit. But otherwise, of course, they're very good for your nutritional value.
Thank you so much, Nisha. Your talk is always evidence
But
based, and there's so much explosion of information that we have. So it's very good that we can navigate through that and know what is evidence based. You talked about this information between the two brains that's going on high speed, sophisticated, and I'm so glad to see that correlation with probiotics, which can help us. Of course, you also mentioned something about the food labels not having the
those
proper labels,
stains.
which I think FSSAI is going to rectify soon. And in your gut dysbiosis slide, please add lack of sleep also.
So that is one of the symptoms of depression, no, or
No, but
MDD.
I
Yeah,
think
but
that
lack of
is
sleep, yes.
also a
Stress,
lot of studies are now showing
definitely,
that
definitely.
that is one
So
of the reasons.
interestingly, we do have a study with. I mean, I didn't have that time. So, but we do have a study with LCS where they have done a study on national medical students. They are undertaking their fourth year, and what was done was they were given the probiotic for 8 weeks before the exam. And what was observed was that the cortisol levels were reduced, their sleep patterns were better, and the non REM sleep, which is the deepest sleep, was much better in the probiotic group. So we do have those studies also. No, so what happens is when you know that you have an exam about 8 weeks later, your cortisol levels start rising and they're maximum just before the exam. So, what they again had, it was a placebo controlled trial. So, what they saw was that when they gave the Yakult, the cortisol levels rose, but not as high as in the placebo group. And they have done 8 week studies, they've done 12-week intervention studies. They have compared the stress markers before and after the examination, and they've always seen that the LCS group benefited better than the placebo group. So in this, it was 80 billion, 80 billion. So you can give three bottles, or like in Yakult we have only 6.5 billion, but otherwise in other countries we have 40 billion, we have 80 billion, we have more numbers also. Of Amanus.
I won't ask.
So there is definitely a dose difference, but that they did help in improving quality of sleep and sleep latency also, which is your ability to fall asleep, which most of us suffer from.
Yeah, right.
So how many bottles of Yakult you recommend in a year?
And so we say 1 to 2 bottles, not more than that. I mean, 1 to 2 bottles gives you the health benefit, but we have seen that for people who have a GI disorder or people who are, low on immunity, for them, more numbers of.
Usually, nowadays kids, do suffer like they're going normal, but during or before the exams, they have some stomach upsets.
Yeah,
They do.
2 bottles, I think, should work.
Yeah, but
No, so the dose, so again, we are not in the drug category. So when we are looking at just health, we are looking at digestive health and we're looking at immunity. So we say 1 to 2 bottles is good enough. But the minute you want to, get, then you kind of increase numbers. Yeah.
Nisha, one question. Any specific time period for intervention for this probiotics when you were sharing the study?
Consuming the probiotics. So we say, so basically, again, another important fact here is that any probiotic bacteria, it's a transient colonizer. So it doesn't become a part of your established part of your gut flora. So they get passed out with the stool. So regular consumption is very important. And because regular consumption is very important, we say have it after breakfast so that you don't forget your daily dose of the probiotic.
yeah, so I think Nisha,
Along with this, it is very important not to have processed food
Hmm.
because it will have negative effects.
Yeah.
Even if you are taking good bacteria, And if you are taking processed food along with it, it will negate the effect.
So the
Whatever
mantra is a balanced diet, physical activity, good sleep, lack of stress for all of us.
New research has come up, activity also.
Yeah, yes, increases the good bacteria and also stimulates your immune system. So yesterday we had an admin party and I think I had something which has touched my throat as a result of it. I'm sorry about that, but yeah.
If somebody forgets to take the food in the morning with breakfast, so is there any time?
Anytime during the day, anytime. But just to remember to take it, you need to have it after breakfast.
So while we are talking about timings, I think although it's not related
Yeah.
to the subject, I just wanted to ask you when you're taking antibiotics, I think
Yeah.
you should take the probiotic 2 hours with a gap
So
of
basically, what we say is that within a gap of 3 hours, you can take the probiotic because it could kill the. The antibiotic could kill the probiotic bacteria.
How many numbers and what we should consider when
So basically, 1 to 2 bottles is very good. When we talk about your 6.5 billion, but 1 to 2 bottles are perfectly fine. But if you're wanting to treat a particular disorder, then you can. Yeah. So we have done studies on IBS. It's a small open trial also where we saw that one bottle was good, just in improving some symptoms of bloating, little bit of diarrhea, and all of that. So anything between
Thank
1
you
to
so
2.
much, Neerja. You always a sounding board for me because of the sound evidence you always provide. So thank you so much.
Thank
First
you.
told me that I have to move on. I think the question.
मैं तो कुछ बोला ही नहीं.
But you'll have to give us.
Yeah.
Anyway, I put those questions to the next speaker because I'm a common man, so I'll ask very fundamental questions. I'm not sure whether yogurt and curd are same or not. Like those questions I want to ask.
So these will be taken up in panel 2, sir.
Yes,
And
the
we have
panels-
ample time and Dr. Neerja is going to stay with us.
Be ready. I'll ask you those questions.
Yeah.
I'll ask whether bacteria sleep or not. I will also ask whether any drug can put the bacteria into sleep. And then I will ask whether before surgery I should take probiotic. And I'll ask question, how to know, this is a serious question, how to know that this guy is having less of probiotics, less of
Good
bacteria,
gut or
good bacteria in his gut. Because it's only we are going through clinical year thinking that he might be having normal bacteria inside and going on supplementing. That would be more scientific questions to me. And just to inform in this way, this contest is as a GI surgeon, when we operate on the patient, we disturb the intestine like anything. We cut, wash with antiseptics, we join, and in the post-operative period, a lot of time to start. Whether during this period, what happens to the microbiota of the gut, nobody knows. When the patient remains in this state, on top of that, we go on pouring antibiotics to this people. And I have seen most of the time the junior dietitians, they used to come and they mainly focus on whether the anastomosis is okay, gut has started functioning or not, how to start the nutrition and how to end the nutrition. But my suggestion is they should keep in mind how many days the patient is having antibiotics, what is the situation of the gut inside, and whether we should give prebiotics, probiotics to these people. They have to come up with an idea because most of the time we forget what is happening. We focus on the overall outcome of the patient. This is a very important part. You have to give me an answer during the session that how in the post-operative period, when exactly to supplement the
Very, very pertinent question. So we definitely look forward to
So
responding.
panelists, please be prepared for
Ma'am, before we move to our next speaker, I would request Dr. Parmeet to present a token of thanks to Dr. Neerja.
very privileged to invite the next speaker for this session, and she is none other than Himani Puri, and she's the Chief Dietitian at Apex Wellness Hospital. She's come all the way from Nasik. Welcome, Himani. She's the Chief Program Officer, and she has a very diverse portfolio. You can see on the screen soon. She is also the part of the core group, the IAPN core group for renal nutrition, and I can see her interest also in sports nutrition. So I think very, very diverse, and we are waiting to listen to you. And you're going to talk about chronic kidney disease, pre and probiotics in chronic kidney disease. Very important for us. So we look forward to your presentation. Welcome.
Thank you so much, ma'am. Good afternoon, everybody. I hope you have a microbiota-licious day today. A lot of information about microbiota and the gut flowing in and around here. Thank you to the organizers for inviting me. Thank you to the esteemed chairpersons here for giving a warm introduction. So I'll be discussing majorly about the probiotics or the gut microbiota where the kidney diseases are concerned. So a lot of information has been flown, and we would be seeing more information about the gut here. So we all know that the gut microbiota is established as soon as the child is born, right from the way the birth is, whether it's through a cesarean section or it's a normal delivery, and it goes on and establishes for the first 2 to 3 years of life. It is the quality which is established in the first 3 years, and then we see an expansion in the quantity of the gut microbiota thereafter. Okay. So the composition changes and improves, and diversification occurs during the entire tenure or during the entire age of a person. As ma'am said earlier, there are almost more than 10 trillion microbial cells present, and we have knowledge only about just 1/8 or 1/16 of the microbiota as of now. What we are discussing is not much, just 1/8 of the entire microbial population in the entire human gut. Dietary habits, of course, play a major important role in how our gut microbiota develops and it grows. So bacterial community is basically a combination of 3 major groups: the bacteriotypes, the formicides, and the actinobacteria. The composition of the intercellular microflora depends upon the region that we are coming from, the family that we are coming from, and the diet we are sharing. So the gut microflora of people in Asia would be different from the people in Europe, would be different from people in the Australia and in USA, because the ethnicity and the environmental conditions also play a very major role in how the gut microbiota flourishes and develops. And with aging, of course, it changes. So, but before we discuss about the probiotic part, there is something else we need to understand about is the prebiotics, which forms the basis of the microflora that we're talking about, right? So, pre is basically the food on which the probiotics flourish, thrive, and give us the results. So these prebiotics basically are the indigestible carbohydrates which are present in the food that we are consuming and provide the substrate for this microflora to grow and flourish. And then comes the probiotics, which give us the beneficial health, which improve our immunity system, which support our gut health, which are involved in the respiratory system, in the urinary health, and in the mental health, as well as ma'am told about the psychobiotics. And together, these pre-probiotics form what we call as the proteobiotics or the postbiotics. So what are proteobiotics or the postbiotics? They are basically the combination of the metabolites of the probiotics. It could be only the metabolites, or it could be the probiotics and the metabolites together. And this entire system of the pre and probiotics is what we call as the symbiotics. So now when we are talking about the renal patients, we do not give them only the probiotics or the prebiotics. We prefer to give them the symbiotics, a pre-probiotic combination, Because even if I'm giving a probiotic, but if there is not good enough bacteria fiber present, then the probiotics would not be able to flourish. And therefore, it is very important that we talk about the prebiotics also. So prebiotics is the fuel for your bacteria. Probiotics is the factories which produce the metabolites, and postbiotics are basically the good which is delivered finally after the postbiotics work on the prebiotics. So where the CKD is concerned, there's a lot of interconnection between the intestinal dysbiosis, the hypervolemia, the systemic inflammation, the myocardial stunning, and the malnutrition which we see in the CKD patients usually. The basic probiotics come into two categories. One is sacrocytic and the proteolytic. Why this is important? Because for a renal patient, we see a lot of uremia happening. And therefore, it becomes very, very important that we understand how the probiotics are working, how they produce the metabolites, and how it is going to affect the metabolic system of the person who is suffering from CKD. So sacrolytic bacteria, they are basically thriving on the carbohydrates, and the proteolytic bacteria are dependent on the protein. Basically, they are the protein fermenters. So when we are talking about CKD, I do not want the proteolytic bacteria. Why? Because they would be producing more of uremic retention substrates. As does the patient is already in a uremic condition. And if I have the proteolytic bacteria which are going to increase the uremia, the patient is going to deteriorate further in the health. So sacrolytic bacteria, they are considered to be the healthy bacteria. They try to balance the microbiota of the colon and is more prevalent in the bifidobacteria and the lactobacilli. They on hydrolyze produce the SCFAs, which basically are the more required substrate or the byproduct of the fermentation of the prebiotics. Whereas the proteolytic bacteria, they basically are protein fermenters. That is, they are going to be increasing more putrefaction. As a result, as I said, we see more of the metabolites like the phenols, the ammonia, the thiols, indols, which are all a big no where the renal patients are concerned. The major species involved are Clostridium, Bacteroidetes, Enterobacterium, Bifidobacterium, certain species from Lactobacilli, which are basically metabolizing the amino acids and forming the phenolic compounds. So this is how the dysbiosis in the renal patient looks like. So when dysbiosis or imbalance exceeds, there is excess of the production of the URS, that is the uremic retention substrates. As a result, we see a lot of uremia in the patients. As a result, we see a creatinine level increase, we see a blood urea level increase in the patients. So a lot of crosstalk is always happening between the organs in the body. Ma'am discussed about the gut-brain axis, how the gut is influencing the brain and how the brain is further influencing the gut. In the same way, the gut and the kidneys are also talking. As you can see here, so what happens is during hypertension, during obesity, during diabetes, which are the comorbidities usually accompanied with CKD, we see an increase in uremic toxins. Nephroprotective molecules are reduced, gut permeability increases, inflammation increases, oxidative stress increases. As a result, we see further microbiota dysbiosis. As a result, this uremic toxins increase further and the circle goes on. What triggers this further is reduced intake of the renal patients. We know that patients who have a high creat level or who are in a uremic situation, they do not feel like eating. They usually complain of nausea. As a result, they end up eating lesser what is being advised or prescribed. As a result, the fiber content also decreases. That means the prebiotic levels or the consumption decreases. And therefore, even if I'm going to supply them or supplement them with a probiotic, since there is lack of fiber, we would not be getting the required results. Colonic transit is decreased. Urea influx is increased because a lot of medications, polypharmacy happens in renal patients. A lot of antibiotics are being given. lot of nephroprotective medications are be giving. The iron levels are low, so we have to balance them also. We have to supply them with erythropoietin sometimes. Plus, if the patient is diabetic, we have to put the patients on OHAs also. For antihypertensive medications also have to be advised many a times. So this all increases the gut permeability, the uremic influx increases, and therefore we see an increase in the proteolytic bacteria. So again, an elaborate explanation of the same thing, how diet becomes the center of everything. Because of diet, the uremic toxins increase. Though tryptophan is considered to be good for relaxing the mind, but tryptophan actually on metabolism is going to increase the inosine PS crescil, which we call as, which is again a byproduct or a uremic toxin. So phenylalanine, tyrosine, tryptophan, choline, lecithin, they all are affected. As a result, we see increase in endotoxins, proteolytic activity, which can further result in renal fibrosis, erythroclerosis, reduced cognitive function, increased neuroinflammation, increased cardiovascular diseases, and probably we might see a lot of immunocompromised patients also with recurrent complaints of fever or viral infections coming to us. So this was one study done, and it clearly demonstrated that in patients with CKD, there was a lot of compensatory mechanism where the gut microbiota was concerned. And what was done was they tried to assess the eliminated products and see how the gut was involved or the colon was involved. And a clear relation was established between a compromised gut or compromised microflora and the renal functions. So again, uremia is going to increase uremic toxins further, which is going to increase protein wasting, dysregulate the immunity system, increase cardiovascular disease chances, and progress the CKD further. This was a small study done where a short-term consumption of diet was done and a comparison was done between the animal sources of the food and the plant sources of the food, which I'll be talking about further in my consecutive slides. And it was found that the animal-based diet increased the abundance of bile-tolerant microorganisms and decreased the levels of formicides that metabolize dietary plant polysaccharides. So therefore, we saw a clear increase in the proteolytic bacteria when an animal-based diet was given to the patients. So clearly the action of the probiotics differs with the diets that is being given. With the animal-based diet, we see a decrease in the bacteriocytes and an increase in the formicides and the proteobacteria, which is the proteolytic bacteria. With the sources of protein, again there's a decrease in the formicides and increase in the biophilia and the bacteriocytes. But with the carbohydrate support in the animal-based protein, there was increase in the lactobacilli and the bifidobacterium, which is the saccharolytic strains of the probiotics. Whereas in the plant-based diet, a more improved or a better growth of the bacteria was seen. Also, we need to understand that the CKD patients also have a compensatory excretion of the products. As a result, the phosphorus levels are also high. So plant-based products help us reduce the phosphorus levels, whereas the animal-based products increase the phosphorus level. So on the whole, where the gut is concerned, where the phosphorus is concerned, and where the metabolic acidosis is concerned, the plant-based diet helps us to manage the renal patients much more better and in a efficient way. So differentiating between the plant and the animal-based diets, high-fat diets have shown to induce an increase in the abundance of formicides as compared to the low-fat diet. The linoleic acid, mainly coming from the plant source, is better utilized by the gut microbiota and helps us to produce the CLA, which is shown to have anti-inflammatory, anti-adipogenic, and anti-diabetogenic and anti-carcinogenic properties. Also, unlike the MUFAs have shown to have inconsistent results, and therefore there was in fact a study done which said that a diet which is high in the MUFA actually decrease the level of the microflora in the gut. So Western diets are rich in SFAs, low in antioxidants, phytosterols, and other phytochemicals, and therefore alter and make the gut more pro-inflammatory. So probably we should be looking at traditional way of consuming food, not with a lot of oil, spices, and salt, but traditional food which has minimum processing and is much more towards the natural process of cooking. So a lot of research has been done on the probiotics, but nothing majorly has been done in the renal patients, especially in the dialysis patients. We usually do not recommend the probiotics or the synbiotics in the dialysis patients because again we lack the data thereafter. Okay? So microbiota modeling in CKD patients has reported that hemodialysis in comparison that providing them even significant amounts of dietary fiber has shown to improve the microflora, but again there is lack of long range studies which are evidence based. Also in CKD we know there's altered protein assimilation, and as a result there is decreased amount of available carbohydrates in the large intestine, which tends to promote the saprophytic microflora, and therefore probably we should be focusing on providing the patient the food which is going to promote the promotion or growth of the saprophytic bacteria, so as we are able to manage the ammonia levels, improve the pH levels in the patient, and also help improve the metabolic acidosis. So when you talk about the microbiota management in renal patients, prebiotics, probiotics, postbiotics, synbiotics, and various diets play an important role. So we now know what is prebiotic, sorry. It's a basically fiber which is not digested by the humans, and basically the gut microflora digests it. They help us to improve transit time. They increase the fecal weight. They help us in calcium absorption and improve the immune function. The probiotic should be something which is resistant to the stomach acid and digestive enzymes, should be fermentable by the gut bacteria, and should be able to nourish or improve the activity of the gut bacteria. So sugar is a carbohydrate, but it does not fall under the category of the prebiotics, right? Because sugar it does not form the substrate for the thriving or nourishing of the microflora. So this is a list of the various studies which were done about the type of the prebiotic which was used and the results in the renal patients. As you can see here clearly, we did show some improvement in terms of the uremic toxins being reduced, the creatinine levels were improved, the GFR function had improved in the patients. But again, these are short-term studies. A very few studies have been done on dialysis patients, and again, very, very few on peritoneal dialysis patients. I would not be going into details of all the studies listed here. So this is a list of the prebiotics which are naturally present in the foods, and maybe something which you can try to prescribe or advise for your patients. But of course, it would depend upon the potassium levels and the protein content of your diet as well. So you have the fructans, which is coming from the wheat, honey, banana, barley, tomato, and rye. Isomaltulose coming from honey, sugar cane juice. Xylo-oligosaccharides coming from the bamboo shoots, fruits and vegetables. Raffinose coming from the legumes, lentils. Soybean oligosaccharides, lactulose, enzyme-resistant dextrin, and oxalo-oligosaccharides coming from the wheat bran and from the whole breakfast cereals, of course, but not the sugar-coated ones. This is a small list of the commercially available prebiotics in the market. I have not mentioned the brand names here, but then you can get them in various brands under various brand names. So all of these can be prescribed with a probiotic if you do not have a combination of a synbiotic at hand available. So supplements in terms of prebiotics, yes, are helpful. We can give them, but the major source has to be diet, because usually renal patients are restricted with fluid intake. Prebiotics are basically fiber. So if I am giving a fiber but I am restricting the fluid intake, it is going to, the patient is going to land up into constipation. So the prebiotic has to be balanced with the fluid intake. So that has to be checked. Also, we have to be looking as I said, about the potassium level, because most of these come with the high potassium levels also. So therefore, that needs to be taken into consideration. Usual recommendation is 3 grams of short chain oligosaccharides and maybe 8 grams of mixed or short chain or long chain inulin, which can be advised to the patient. Next, I come to the probiotics. So probiotic was already spoken and discussed by ma'am, that they should be non-pathogenic. They should benefit the human beings, right? We cannot term any microorganism to be a probiotic, okay? They should preferably of a human origin. They should be or capable of surviving and growing in vivo. They should be able to endure to the low and the high pH concentrations, should be non-allergic, and should not be mutagenic or carcinogenic to be defined as a probiotic. So this again is a list of the probiotics which have been prescribed, used and advised in the renal patients. Again, these are short-term studies. We do not have long-term studies to actually come to a consensus of how much prebiotic, probiotic can be given. There is no prescription as such about the recommendations in the renal patients again. So growing evidence highlights that we can prescribe the probiotics, but we need more guidelines to regulate the advices or the prescriptions. Normally, thermophilus, acidophilus, coagulans, and longum have been shown to have good results in CKD patients and shown to improve the uremic levels in the CKD patients. Next, we come to the postbiotics. So postbiotics basically would be either a combination of a probiotic and the byproduct, the metabolite, or maybe just the metabolite. They help us reduce the risk of microbial translocation, infection, and improve the inflammation response of the body. Again, they are promising, but we still need to explore them further. Certain examples include yogurt, sauerkraut, miso soup, soft cheeses, kefir, sourdough, buttermilk, pickles, and tempeh. But again, these all come usually with a high salt level and a high potassium level. So most of them actually go out when the renal patients are concerned. A study talking about the symbiotics, which was studied in the renal patients, and there were promising results. A couple of studies did show us improvement in the reduction of the CS levels and the reduction in the inosyl sulfate levels also. Of course, with all this comes the dietary modifications with the renal patients. So in the renal patients, we try to reduce the protein intake so as to reduce the uremic toxins, so as to reduce the effect of the proteolytic bacteria, or we try to combine a diet in low proteins with a keto analog so that we are able to avoid the muscle wasting and try to balance the gut health of the patient as well. So a lot of positive effects were shown when the patients were put on a low protein diet or a VLPD with the keto analog or on the plant-based diet, which showed us a good improvement in the renal parameters of the patient. So effect of the natural and artificial sugar on the gut microbiota. With the glucose, fructose, sucrose and lactose, there was increase in the bifidobacterium. Artificial sweeteners actually tend to decrease the growth of the bifidobacteriums. Bacteriocytes again were reduced in the natural sugars and improved in the artificial sweeteners. So whatever was not required was actually improving or growing with the artificial sweeteners. Decreasing with the artificial sweeteners. So maybe I'm not advising artificial sweeteners, but it's a possibility to say that we need to be reducing the natural sugar intake here. Yes, ma'am. I'm just finishing up. Okay. So, a comparison between the normal and the CKD population. CKD patients have fewer good fewer good microflora, and they have more bad gut microbiota as compared to the healthy patients. That is what the focus should be on when you're talking about diet planning here. We could be looking at a high fiber diet and a diet which is not advising a diet which is high in protein and not advising diet which is high in the fat. Mediterranean diet helps us to improve microbiome diversity because it is going to be majorly based on dietary fiber. So prebiotic is going to help us improve the gut microbiota. So the foods which are going to promote dysbiosis and the foods which are going to actually help us maintain a symbiotic relation with the genomes. Again, to conclude, residual supplements or the resident starch diet is going to again improvise or help the gut microbiota. Thank you so much.
Thank you so much, Shivani. And the very important point at the end of it is the shift in the approach to CKD patients from the animal-based diet that were earlier being prescribed when we were studying to a plant-based diet
Correct.
now. I work with renal patients, so I have many questions, but I don't want to make that folly again. Dr. Dash, you want to ask any question before?
But since it is time, I would some questions reserved for the panelists again. One is whether we should always mix pre and probiotics. If it is there, then so whether we should mix pre and probiotics all the time while treating the patients. If so, then what doses, what proportion? Second is what about the alcoholics? Those who take alcohol, whether their gut bacteria is destroyed or not destroyed, and if so, then what should it be? That would be two questions to know. And of course, it was a very enlightening talk. These three, I had no idea about the postbiotic one. I think the postbiotic things we need to discuss more and more. How to put it into clinical practice. Thank you.
I think
Thank you.
Thank you so much, sir. Thank you.
Postbiotics will come as drugs very soon.
Yes.
So I think we'll.
So in
wait
fact, PMT is kind of coming into trend as a postbiotic because we are talking about the later management of the treatment. So so probably FMT should be the fecal microbiota transit could be in the postbiotics.
So right now, I don't think we have a recommendation for a particular strain of bacteria to be given to CKD patients, or we don't have particular probiotic or prebiotic recommendations. But what we can do with the diet is something that is open for all of us. Thank you.
Yeah, in that connection, one very important question. I just remember where I used to discuss that question. A lot of people do not have answer to that. Maybe a friend from Japan would be able to answer. Is what happens to these bacteria when the patient is immunosuppressed? Because we know that when you are immunosuppressed, even your normal bacteria would become pathogenic. So whether we should, and we know when they are immunosuppressed, we give lots of antibiotics. If that is the thing, then whether we should supplement, or if we supplement, they would become virulent. We don't have any answer. I have no answer. Maybe that would be one thing to search for. There's a lot of things are coming up which you can note down for future research.
So just to add here, I mean there are fecal tests to check your gut microflora, but then it is not very common here in India. We do not have access to it because it requires a genetic testing also, which is just not a fecal sample test. The fecal sample is sent to a gene testing lab where they are testing for the microflora present in your gut through the fecal sample. But unless we do that, we would have no knowledge about what is existing in the
No, what the
gut.
fear is if I supplement bacteria to a person who is immunosuppressed and on antibiotics, whether that bacteria is going to become pathogenic or
No.
not pathogenic. And in this connection, I am to inform you that there is a very peculiar thing. See, we are all worried about the resistance. that the bacteria are developing resistance, the killing bug, and so many things. But nothing is heard about the resistance of these bacteria, which are normally there in the gut. So we are taking, if they become resistant to antibiotic, job is finished. So that's not happening. Why that is not happening? That's an area of research again.
So, I mean, post-transplant, we usually do not advise for curd. I mean, we don't say no for curd or something or yogurt per se. So, I think natural sources of the probiotics the patient is consuming.
But in liver transplant, protocol, whenever the patient starts, take plenty of curd.
That's
Yeah,
what we
I
are
have no
actually
idea about
giving.
the kidney, but I can
Even
tell
for
you in
kidney
liver,
patients,
We do.
provided
The
it is
potassium.
safe and it's sort of served in a hygienic
Correct,
manner.
correct, right. That's what. Yeah. So again, I mean, supplementation commercially or through food, that
Yeah,
also
so
needs to be explored maybe.
right now, yes, it's better to stick to foods because we
Correct.
still don't know what the
we
recommendation.
have a long range of immunosuppressed people, severely diabetics, the cancer patients, the immunosuppressants for transplant, anti-cancer therapy, congenital problems. So in those cases, we need to explore what is happening to these things.
Yes,
Thank you.
immunocompromised patients are a different ball game, and I think we have to be very cautious with them.
Yes.
So
Thank you
Thank
very
you so
much.
much.
anyway,
Yeah, ma'am. Ma'am, you want to begin?
I just wanted to ask that you shared there was a small study for fats in the increase
So again, as I said, it was just one study which was done,
yeah,
and yeah, so and they just categorized it that there was a decrease in the microbial growth. That's all. There was no categorization about the proteolytic or the saccharolytic or the good bacteria or the bad
because
bacteria.
otherwise it will change our ratio of mufa.
Exactly.
That also because there is a shift from animal to vegetable protein first
Correct.
of all, and then there will be shift in the ratio.
Right.
So thank you so
Thank
much.
you.
Himani.
Thank you, Himani. I would request Nilasha ma'am to present a token of thanks to Ms. Himani Puri.
I would also like to ask the panelists what happens to the curry. Curry, we are taking curry means you have to boil the curd and make it. What happens to that? If you have any idea or not. So moving forward, it's my pleasure to request or invite Professor Saurabh Kedia, our faculty from the Department of Gastroenterology. Please be here first and then I'll come. Please be here. And see, he is going to deliver a talk on the gut microbiome. Initially, I was supposed to speak on this. Then I said I'm the least person to speak about this. I thought about a lot of people, and then I zeroed in on him. And very short notice, I requested him that you are the best person to speak on this. He is a practicing gastroenterologist, basically from Gorakhpur, a product of AIIMS Delhi. But he can manage everything from esophagus to rectum very well. But his focus of interest is intestine. Those of you who are looking at the internet regularly, searching for this, you'll find his name almost many places. So a lot of research done, lots of presentation nationally, internationally, and has got awards also. And presently he has many projects on this. He is from the Department of Gastroenterology and Human Nutrition. So I think he's the best person to speak on this aspect here. I invite Dr. Saurabh Kedia
to speak on his. Yes, thank you.
Good afternoon, everyone. Thank you, sir, for this kind and exaggerated introduction. And my job is to talk about the probiotics and the gut health. And my sincere thanks to the organizing committee also for this opportunity given to me. So many sections in this talk would be a repetition, but I think that it would be a good revision for you all. So I'll start off with the discussion about the gut microbiome, and then in the later part of the talk, I'll discussing about the role of probiotics in the gut health and the GI disorders. So first of all, what is microbiome? So basically, there are two terms: microbiota and the microbiome. So microbiota basically is the collection of entire organisms in a given sample, which can be gut, stool, saliva, any sample. And microbiome is the collection of those organisms and their genetic content and the surrounding environment. So it's basically what it tells is who are there. So there are several other terminologies related to the microbiome, like the metabolome, the metagenome, the metaproteome, the metatranscriptome. But to understand in a very basic manner is that who are there is denoted by the term microbiota or microbiome. And interestingly, microbiota is not only there in the gut, but it's there throughout the body, in the oral cavity, in the esophagus, in the stomach, pancreas, liver, gallbladder, small intestine, large intestine. Every area of the body has the microbiome, and it has been characterized through various technologies and the advanced genetic and the next generation sequencing. Of these, obviously, gut is the reservoir, has the maximum number of these microbiota, and 95% of the body's microbiota is found in our intestine or in our gut. And in the gut also, the maximum amount or the maximum concentration of the microbiota is found in the large intestine. So man is a superorganism. We are not only we, but we are also influenced by our microbes. So we are we plus our microbiota. And interestingly, the number of microbial cells which we harbor and their genes is almost equal or maybe more, even more than our human cells. So the number of cells and the number of genes which the microbiota has is equal to or more than the number of cells. And if you look at the variation, Dr. Puri already discussed about it, that our genome is 99% similar, but our bacteriome is not. So every individual has a different microbiome. And unlike the genome, so which is one of the very important confounders when we analyze or we do any study related to the microbiome in humans. Now, if we look at the composition of our gut microbiome, then again discussed previously that there are 6 major phyla of the gut microbiome: the Firmicutes, the Bacteroidetes, Actinobacteria, Proteobacteria, and the 2 rare are Fusobacteria and the Verrucomicrobia. Of these, the most important, the most common are Firmicutes and Bacteroides. And interestingly, the ratio of Firmicutes and Bacteroides varies from 95% to 5% in one individual to 5% and 95% in the other individual. So overall, they may be 90% of the entire microflora, but again the ratio varies, which again highlights that the microbiome is very individual specific and very different from other person's microbiome. So now, what does this gut microbiota do? What does this healthy gut microbiota do? It has lots of functions which we cannot imagine. So it has functions related to the gut, and it also has functions outside the gut. So if we look at the functions in the gut, then the healthy microbiome, what it does is it inhibits the bad bacteria. So it competes with the bad bacteria for the place as well as the food. So there is competition for the place where it lives, and there is also competition for the food which it eats. Again, it acts on the intestine, it educates its immune system, and it also maintains the epithelial barrier so that it prevents the translocation of the harmful bacteria and the metabolites from the gut into the human body. It also has several metabolizing functions, which include the biosynthesis of vitamins and amino acids. It metabolizes the drugs. It breaks down the food. So there are saccharolytic, the proteolytic bacteria, which can break down various components of the food and release harmful or the good metabolites, depending upon the individual's composition of the microbiota. Again, as I said earlier, it provides resistance to the pathogens. It protects against the epithelial injury, and there are several axes. So microbiota can influence the brain, it can influence the bone, it can influence the kidney, it can influence the adipose cells, it can influence the heart. So there are several functions and influences of this microbiome in our human body, which is being realized day by day. So every day we have one new information on the microbiome and its role in the human body. Again, a simple representation of what does the microbiota do. So it provides resistance against the pathogens. It digests the undigestible dietary the fiber. It synthesizes useful products and the metabolites for our body. It synthesizes many useful metabolites such as the short chain fatty acid, the secondary bile acid, which again are useful for our body. It metabolizes several drugs, and it also maintains the epithelial barrier and provides the immunotolerance. So now again, several interconnections between the microbiome and other parts of our human body. So there is a strong connection between the liver, the gut-liver axis, and both liver and microbiota influence each other, and it has a significant role in maintaining a healthy liver as well as in patients with different types of liver disease. Then we also have the gut-brain axis, which has been discussed earlier, the gut epithelial barrier, as I discussed, and also the gut adipose tissue barrier, where it influences the insulin resistance and the fat metabolism in our adipose tissue. So now microbiome has several influences. What influences the microbiome? What decides what microbiome should a individual have? So it starts off with the birth. So genetics composition has influence on the microbiome, and up to 10% of the microbiome is heritable and may be influenced by the genetics. Then it starts off with the delivery, and before delivery also the maternal lifestyle, the maternal habits can also have some influence on the infant's microbiome. And then again with the mode of delivery. So patients who are born vaginally have a different microbiome. Patients who are born with the cesarean section have a different microbiome. So vaginally delivered babies have a healthier microbiome than the cesarean section delivered babies. Then the mode of feeding. So patients who are breastfed have a good microbiome because the breast milk has several compounds, the oligosaccharides, the prebiotics, which influences the microbiome and leads to increased growth of the bifidobacterium. The formula-fed infants will have a different microbiome. Then as the child grows up, he gets exposed to several environmental factors such as the diet, the environment, the geography, which influences the microbiome. And by the age of 3 years, the microbiome reaches the configuration of that of the adult. So till 3 years, it gets influenced, and by 3 years, it becomes stable or it gets stabilized. Then, as discussed earlier, also the geography. So the rural individuals have a different microbiome. The urban individuals have a different microbiome, and it's been shown that the rural individuals, because of their lifestyle, their habits, their diet, have a healthier microbiome. Then one of the very major influencer is the diet. So again, as discussed earlier, the plant-based diet, the animal-based diet. So high sugar, high fat diet will have a detrimental effect on the microbiome. The plant-based diet, fiber-rich diet will have a good effect on the microbiome. Antibiotics. So even a single dose of antibiotic can affect your microbiome, which can last longer. So antibiotics have a major negative influence on our microbiome, and again, travel can be one other factor which does influence the microbiome. So now, what is the food for the healthy microbiome? So as shown here, again, plant-based healthy diet rich in fruits, vegetables, fresh fruit, whole grains will have a positive influence on the microbiome. Intake of processed food, red meat, refined sugar will have a negative influence on the microbiome. Now, once we have discussed about what is a healthy microbiome, what is dysbiosis? So we define dysbiosis. Then dysbiosis is abnormal microbial colonization of the intestine, where changes in the quantity and quality of flora become pathological and harmful. So there are two terms, quantity and the quality. So by quantity, what I mean is that there is decreased abundance and diversity. So what is diversity? So like this hall has a very low diversity because all are dietitians. So if there were dietitians, cardiologists, nephrologists, physicians, engineers, IT people, then it would have a very higher diversity. So the healthy microbiome is diverse. There are different types of microbiota. So different type of barbecue, different type of bacteroides, different strain, different species. So it makes it more healthy and more diverse. Whereas dysbiotic microbiome is less diverse. There will be very few species. And then in terms of composition, so firmicutes and bacteroides are the healthy bacteria. They produce healthy metabolites. So their quantity will be higher in patients who have a good or a healthier microbiome. Whereas patients who have disease will have increased abundance of proteobacteria, which are the harmful bacteria, which are proteolytic. So this is basically what is dysbiosis, the quantity and the quality. And now again, what causes dysbiosis? So we have listened that dietary fiber, vegetarian diet, breastfeeding, vaginal delivery will have a positive influence of the microbiome. Whereas antibiotics in the child as well as in the mother, Western diet, drugs, infections, stress will have a negative influence on the gut microbiome. And what does dysbiosis do? So if the microbiome is healthy, there would be increased healthy metabolite production like short chain fatty acids, increased antioxidant capacity, decreased inflammation, improved metabolism, increased better insulin sensitivity. And if there are factors which influence the microbiome negatively, such as the diet, the drugs, what will it do? It will decrease the healthy metabolites, it will decrease the short chain fatty acids, it will decrease the secondary bile acids, it will increase the harmful metabolites, lead to increased abnormal inflammation, and subsequently the disorders which have been associated with dysbiosis. So now, what are the diseases which are associated with dysbiosis? Again, as we are our knowledge is improving and we are becoming wiser, almost every disease is being linked with dysbiosis. If you look at the GI disorders, the disease which have a very strong correlation with dysbiosis are the inflammatory bowel disease. So it's a type of disease which affects our intestine, whereas there is inflammation, formation of ulcers, narrowing in the intestine, and the patients can present with abdominal pain, diarrhea, weight loss, bleeding per rectum. So the common disorders are ulcerative colitis, Crohn's disease, and pouchitis. So it does have a very strong association with dysbiosis. Then we have functional GI disorders such as irritable bowel syndrome, constipation, and the gamut of these disorders, which again have association with dysbiosis. The liver diseases like alcoholic liver disease, metabolic liver disease, the fatty liver disease, patients who have complications of cirrhosis, such as patients who have encephalopathy, who have ascites, will again, and other disorders like primary sclerosing cholangitis, again have been associated with dysbiosis. The acute diarrhea, the antibiotic associated diarrhea, and Clostridioides difficile colitis have a very strong correlation, and the cause of these disorders is the antibiotics, which has a negative influence on the healthy bacteria, and because of this, these harmful bacteria grow up and they can cause antibiotic associated diarrhea or Clostridioides difficile colitis. The metabolic disorders such as the metabolic syndrome, obesity, diabetes have also been associated with dysbiosis. And if we again look at these axes, then the gut-brain axis can have influence on the neurodegenerative disorders such as Alzheimer's disease, Parkinson's, multiple sclerosis, and then skin disorders such as psoriasis, atopic dermatitis have been associated with dysbiosis. Metabolic disorders I already discussed, and the gut link axis has also been talked about. And with the advent of COVID, a lot have been discussed about the influence of gut on the lungs and the lung disorders. So now what we have learned is that there is a healthy gut microbiome which has high diversity and a defined composition which is different between different individuals. Then there is a dysbiotic gut microbiome which is associated with several disorders. And now if there is dysbiosis, can correcting this improve the disorder? So now we don't know, like chicken and egg, that whether dysbiosis came first or the disorder came first. But still, we have studies which have shown that correcting the dysbiosis may improve the diseases which are associated with dysbiosis. So how do we correct dysbiosis? So we can give antibiotics. So what will antibiotic do is they will kill the pathogenic bacteria, leading to increased abundance of the useful bacteria. But what would be the side effect? The antibiotics will also affect the useful bacteria. So they may be useful in short term, but in long term they may be more detrimental and may be more causative. So antibiotics are only recommended for short term and in general not recommended. Probiotics, prebiotics I will be discussing. Diet again. So the Mediterranean diet, the healthy diet, the plant-based diet, the anti-inflammatory diet, which is rich in vegetables, fruits, grains, which is poor in processed food, red meat, will have a positive influence on the microbiome. And studies have shown that diet as a therapy can lead to correction of these disorders, especially in patients with inflammatory bowel disease. The use of exclusive enteral nutrition, use of exclusion diets, anti-inflammatory diets has shown improvement in these disorders. Then fecal microbiota transplantation, as was already discussed earlier. I'll be discussing briefly about it in the later part of this talk. And there are other novel modalities such as viruses, phages, which can again have influence on the pathogenic microbiome and can lead to improvement in the gut microflora and amelioration of this dysbiosis. So now again a revision of the glossary on the terms probiotic, prebiotics, and symbiotics. So probiotic is basically a live microbiome which, when administered in adequate quantity, will confer health benefit. Prebiotic is a substrate which would be utilized by the healthy bacteria and will again confer a health benefit. Symbiotic is a combination of prebiotic and probiotic, and postbiotic, as discussed earlier, is an end product of the probiotic. So now a very interesting story behind probiotics. Probiotics are not new; they are more than 100 years old. And who discovered probiotics is Meshnikov. He's a great scientist, a Nobel laureate, who, with his astute observation in 1997, saw that certain inhabitants and certain farmers were living older than the others. And what he found was they were drinking a fermented yogurt drink on daily basis. And then he found that this drink contained a probiotic bacteria called the Lactobacillus bulgaricus. So this was the beginning of the probiotic story. And there are other interesting stories also, which again are responsible for the nomenclature also. So Alfred Nissel was a German professor, and during First World War, what he noted was that there were epidemics or outbreaks of severe diarrhea, and there were some soldiers who were protected, who did not harbor, who had a very mild infection. And what he found was there are certain strains of E. coli which were found in these soldiers. And this led to the discovery of E. coli Nissel, which again is a probiotic formulation. And there are other interesting stories also in the past which have led to this probiotic story and have invented the probiotic concept. And now a bit about the science of probiotics. So what is probiotic? So there are three terms: a live microorganism, adequate amount, and health benefit. So this will be a probiotic. So what constitutes probiotic? What are these live microorganisms? Most important and most commonly used are the lactobacilli and the bifidobacteria, which are commonly known as LABs. Then we have other strains such as the E. coli, the fungi such as the Saccharomyces boulardii, Clostridium butyricum, and certain species of bacilli. It is very important that these effects are strain specific. So even among the species, there are certain strains which will have probiotic effect. There are certain which are not. And based upon this, they also have been named. Then the adequate amounts. So again, the appropriate dosage needs to be delivered to have an appropriate benefit, and this again would be strain specific. So some strain will require more, some strain would require less, and they would have health benefit in the form of maintenance of general health, improvement of general health, prevention of disease, or correction of disease. So now, where do we get probiotics from? They could be probiotic rich foods, food supplements, and the drug formulation or the medicines. So you all must be aware of the foods which are rich in probiotic: curd, lassi, fermented vegetables, cheese, which have different strains of these bacteria. I will not go into the detail. And interestingly, this study looked at the composition and the laboratory correlation of the commercial probiotics in India. So there were 20 preparations which they looked at. They did sequencing of these bacteria isolated from these probiotics. And what they found was that there was very poor correlation between the declared contents on the pack and lab values in the viable cell count colonies, the genus and the species strain identification, presence of contaminants. So what is said may not be there. So if the recommendation was 10 to the power 12 colonies, what was found was 10 to the 0.9 to 10 to the power 10. So we have to be very cautious in prescribing a particular brand or a particular strain of probiotic to our patients. So again, revising what could be the characteristics of effective probiotics. So they should be viable and they should maintain a shelf life in the preparation which needs to be given. They should survive the passage in the digestive system, should colonize in the intestine, compete with the already resident organisms, should be safe. So there should be no antibiotic resistant genes in them and there should be no side effects with them. Should be able to get the food from the normal diet and should have a beneficial effect. A bit about what are prebiotics and symbiotics. So again, three concepts: a substrate which will lead to growth of the microbiota, a microbiota, and the health benefit. So what would be the substrate? The most common substrate are the non-starch polysaccharides, the oligosaccharides, the resistant starch, long-chain fatty acids, and polyphenols also have been recently found to have prebiotic properties. The usual microbiota which gets increased with these are the bifidobacteria and the lactobacilli, and again they should have health benefit. So again, there are several food products which are prebiotic sources, and the most common oligosaccharides are the fructans or the fructooligosaccharides, which are mostly found in food, in fruits, inulin, the galactooligosaccharides, which are found in the milk-based products, the dairy products, lactulose, which is a disaccharide, a common agent used for constipation, also has a probiotic effect. And one of the most important prebiotic which we all have been exposed to and it's recommended is the breast milk oligosaccharides, which have a very positive influence in increasing the healthy bacteria such as the bifidobacteria. So what are symbiotics? A combination of prebiotic and probiotics. So there are two concepts to it. There are two types, complementary and the synergistic. What complementary means is that there would be a prebiotic and there would be a probiotic, but they would be independent of each other. So this prebiotic may increase the growth of some bacteria, but not the bacteria which is there in the probiotic. What are synergistic? So both have interaction between each other. So the live microorganism which has been chosen will thrive on the substrate which is there in the symbiotic. So they have the substrate will lead to increased growth of the live microorganism which is there in that product. And what do they do? How do they correct dysbiosis? So they will have competition with their enemies, the bad bacteria, and they will outperform them. They'll produce antimicrobial metabolites. They'll produce useful metabolites. They'll produce vitamins. They will increase the integrity of the intestinal bacteria. They'll have anti-inflammatory effect. They will produce anti-inflammatory cytokines, and also they will have systemic signals by the production of neurotransmitters and other metabolites. So now, what is the evidence on these probiotics and prebiotics in gut health and the GI disorders, and what do the guidelines recommend? So, prevention of antibiotic-associated diarrhea and Clostridioides difficile colitis. The evidence is strong for this, but only few strain combinations have been found to be effective. But they are effective only in patients with high risk of recurrence. But there is a caveat to this guideline. It suggests that patients who are at risk of adverse events, such as patients who are highly immunocompromised and who cannot afford, can avoid. It won't have major impact if they avoid these probiotics. So again, it means that the effect would be very less and it would not have a major influence in the prevention of these diarrheas. Treatment of antibiotic associated diarrhea and C. difficile colitis, there is no recommendation, so only in clinical trials. Prevention of necrotizing enterocolitis in children, again few strain combinations have been recommended for this indication. Coming on to the GI disorders in adults, ulcerative colitis, there are few studies, one from AIIMS also, which showed beneficial effect of 8 strain probiotic in patients with ulcerative colitis. But again, because of the weak evidence, low quality evidence, the AGA does not recommend it as a routine treatment, only in clinical trials. The other problem with the use is that the study which was conducted in AIIMS used VSL3. But the problem was that the amount of bacteria which was required would amount to 36 capsules of VSL3 per day, which would be impractical as well as not affordable for majority of patients. For Crohn's disease, again, very bleak evidence, no recommendation, only in clinical trials. For pouchitis, which occurs in patients with ulcerative colitis who have undergone surgery, so what is done is the colon is removed, a pouch from ileum is created and it is joined. So the ileal pouch and anastomosis, in patients who develop inflammation of the pouch, there would be some use of probiotics in the maintenance of remission. So in making the patients better. But again, what is suggested is if there are risk of adverse events and cost can be avoided. Acute diarrhea, no recommendation. A recent study in New England Journal of Medicine showed it was not beneficial. So again, only in clinical trials. Patient with irritable bowel syndrome, there are more than 70 studies. Some have shown benefit with some strains, but again, no definite recommendation that a probiotic should be used for IBS patients. And for H. pylori also, there is no recommendation. So we look at the evidence and the science. There is much, and it shows that probiotics have a lot of benefit. But if we look at the real clinical practice, there are very few indications of probiotics in patients with GI disorder. Now, should I prescribe probiotics for the maintenance of health? A person is healthy, he doesn't have any disease. Should he take probiotics to increase his health? In terms of probiotic-rich food, yes. But in terms of supplement and medicine, no. There is no evidence that it will improve the health of an individual. So now, why are these probiotics weakly effective? Because of the heterogeneity of strains, heterogeneity of dosage, heterogeneity of formulations, and single or few strains. So we have 1,000 species in our gut. With one strain, how do we expect that it will change or it will have a major effect on the gut flora? So basically, microbiomes may work better as a team when multiple types of these microorganisms are given, and this brings the concept of fecal microbiota transplantation. So what is FMT? It's infusion of fecal suspension from a healthy individual into the gastrointestinal tract of an individual GI disease. So it would have a better capacity to replace the gut flora. So there are studies in several disorders, like in Clostridioides difficile colitis, it has shown a definite benefit, and it has been recommended by the US FDA as a therapy for patients with recurrent C. diff colitis. In patients with IBD also, it has shown benefit. Although, other than C. diff, as of now, the use of FMT is only recommended in setting of clinical trial. Then we have the concept of next generation probiotics. So what are these? There are newer strains of microflora which have a very selective impact on a particular indication, and they could again may improve the horizon or increase the horizon for the use of probiotics in our setting. So to conclude, probiotics do have a beneficial effect on our gut health, but because of these heterogeneities, the of the preparations of these strains, as of now, they have a very limited role in patients with GI disease. Thank you.
I will say that your presentation was indeed excellent and comprehensive.
Thank you.
And your mode of delivery was lucid and easy to understand by all. So that is also a plus point. And all the topics were well covered. And how to, what is the data and evidence available, that is was also taken care of. And you rightly said that for the individuals now, we don't have to go by population approach or a group approach. We have to take a person as an individual, and it is a time for precision nutrition or personalized nutrition. Along with microbiome, genome is also to be taken into account if we are prescribing diet. So, although it is this approach is in infancy, but now you have sensitized the young minds who are present here, and definitely everybody would like to work on this new field, and a lot of work needs to be done. Congratulations.
Thank you.
Thank you so much for the clarity in terms of the evidence and the recommendation. Very nicely put. And since you work in the Department of Gastroenterology, I was wondering if there's any information about how the gut dysbiosis happens in tuberculosis patients, because then again, so I was wondering, I was curious because we have so much, maybe we'll discuss that later at lunchtime if possible.
So with a lot of clarity, we still have to have a cocktail with lots of things in it. So still we don't have a solution.
Yes, ma'am. So we still don't have. In terms of probiotics for GI disorders, we don't have a magic.
So is there the cocktail formulations, would that be a better approach than having just the single strain?
Yeah, mostly yes, ma'am.
So something like BSL-3, etc., which has a cocktail.
But again, BSL-3, as I said, that for efficacy it requires.
You need such a, and they are so expensive.
Yes.
So thank you so much.
Thank you, ma'am.
Basically, say we are using we have 6 classified things to be used, but question is how to decide where to give prebiotic, where to give probiotic, where to give symbiotic or postbiotic. Okay, understood. And these 3, the judicious decision, actually the recommendations are there. I'm not sure they are not there. Second, also I used to think in place of fecal microbiota transplant, whether you can really give enema of these things to increase the concentration in the gut. These things also are not very clear. Whether in place of them you can infuse per rectally and you will get a better concentration inside, because it is at least this thing is known. I'm also aware that what we're taking in terms of capsules are not reaching there in full concentration which is required, and how only one can replace all the types of mycobacteria inside the, but something is better than nothing. So might be using, there are lots of trials on pre and postbiotics in different areas, including septic conditions in acute necrotising pancreatitis, sepsis. So some are showing good results, some more are saying that we are not very sure with the good, and very few are saying that it's actually not good. So my suggestion is, whenever we are attending a focused session like this, not only where we should be swayed away by the positive things, what is being told here, and a student of science, we should know everything. What is the bad, what is the good, where we are lacking, what is the area of research. I want you to take note of all this. And till now, whatever is being discussed, I'm really very impressed as a MBBS student. So I think you should keep a record of the link which we are transmitting through YouTube, and you can at your leisure again go through this. You will get much more things from inside. In every slide, what was presented was equally important. Thank you. Thank you very much.
Sir, I will request you to present a token of thanks to Dr. Saurabh.
Oh, go to the camera.
In a marriage, I found a person who is everywhere, even if where we were not allowed as guests or something, cameraman good. Here the Pandit by the side of the bride, and we are not allowed. But you, so anyway, in front of the camera. Thank you very much.
I would also make a small request for acknowledging the presence of our chairpersons and sharing such valuable information to all our delegates. I would request our guest of honor, Dr. Vijay Lakshmi, to present token of thanks to all our chairpersons. Dr. Pameet, please.
ये सब जो शादी ये सब बात हो रही है वो YouTube में जा रहा है. Please edit all this.
There are a lot of questions from Dr. Das. One question, that is, bacteria is sleeping or not. So any idea? Any idea from audience? Any evidence? Any proof I require? So bacteria is sleeping. He has forget the growth curve of bacteria. So bacteria only survive for either for seconds, for minutes, for the hours, and for days. So there are different phases. Lag phase, where they accumulate everything for their eating. Log phase, where they multiply. Stationary phase, where no activity, it means they are sleeping. and then decline phase, they are dead, means going. So bacteria are sleeping. Thank you.
My question was whether bacteria go to depression or not.
So before we break for the lunch, I know we need to finish off on the time also. We have such an important day. Apart from IPEN India Day, we have a very, awaited match which has been started between India Pakistan too. And you people might be loving to see maybe the concluding time of the match by the time we are free in the evening. So basically, I want to show a small video because in the beginning I missed out showing about the theme. So this is a small video which actually leaves us with a message that why this theme was been chosen by IAPEN India and what is the major importance of this theme. The medical diagnosis is incomplete without nutrition diagnosis. So I'm just sharing a video. The volume.
Despite India's impressive economic growth over Despite India's impressive economic growth over the past decade, we remain a nutritional weakling. Every survey has reconfirmed the urgent need to address the issue of malnutrition. The prevalence of malnutrition has been estimated to be as high as 50% among acutely hospitalized adults. Despite the relevance and the prevalence of the problem, malnutrition frequently remains underdiagnosed and undertreated. Malnutrition is a leading cause of morbidity and mortality, increasingly recognized as a silent epidemic globally. Nutritional compromise generally worsens during inpatient admissions, with hospital-acquired malnutrition affecting up to 65% of patients, and often do not meet their daily need for energy, protein, and other nutrients. Poor nutritional status can reduce wound healing and tissue repair and lead to decreased immunity and inability to tolerate and overcome stressors like infections, increased rate of complications, and increased healthcare costs. Implementation of nutritional diagnosis and intervention significantly improves clinical outcomes and reduce healthcare expenditure. Timely nutritional care is fundamental to good clinical practice. Nutrition intervention coupled with clinician collaboration is critical in remediating malnutrition in hospitals and to improve patient care and reduce hospital costs. Time has come for integration of medical nutrition therapy with pharmacotherapy, consequential only to early screening and detection of malnutrition in every healthcare setup. You, me, we. So let us improve the nutritional therapy together in malnutrition. IAPEN India proposes the Chennai Declaration. We look forward for your valuable support for this initiative. Thank you.
So this was all about the base behind our theme, why we have chosen this theme as for IPEN India Day. And we all realize that nutrition care process is not something which is very being generalized into the hospital setting. It is something which has been tailor made with for the patient. I mean, individualized for the patient whom we are taking care of. So once we are being aware of this and been following into our institutes, into hospital for taking care of the patients, then definitely the time would be not far when we are being like not far from even from the medical practitioners that we as a dietitian who are the basic stakeholders for carrying this nutrition awareness forward, we can definitely do wonders into it. And IAPEN India has taken this initiative in ICNC 2023. And then thank you, Mr. Shiv Shankar, for sharing this video. And I could share it with you all people over here. And IAPEN India is working onto the nutrition care process where showing the importance of medical diagnosis individualized to different diseases is being taken care shortly. Maybe by next year, we'll be coming out with this. So keep your finger crossed and let's wait for something new been coming up from the association. Thank you so much. We'll be breaking here for the lunch and we'll be back. It's 2:45, 1:45. So we'll be coming back over here by 2:30. The lunch is in the same area outside the this auditorium. And by 2:45, once we reach back, I request everyone to be kindly on time because we need to finish also on time. Thank you so much.
हाँ हाँ
So good afternoon. I can...
So good afternoon, everyone. I welcome you all with the post lunch session. I think it's very difficult. It's like getting the people back from the graveyard to the life. But still, I think there are interesting sessions we try to keep post lunch. So hope it keeps everyone away. And I will like to introduce our chairpersons for the next session. Our first chairperson is Mrs. Anita Jitana. She is Vice President IAPEN India Delhi Chapter, Consultant Dietetic in the Trust Apollo Hospital, New Delhi. Ma'am is having more than 35 years of experience in the field of nutrition with special interest in critical care. She has been awarded for outstanding contribution to the field of dietetics at the 75th World Founder Day celebration. She has won Apollo Innovative Award for Clinical Excellence for Research and also won the award in IDECON for research in role of omega-3 in oncology. We welcome you, ma'am. Our next chairperson is Ms. Anjali Sharma. She's the chief dietitian with Maharaja Agrasen Hospital, New Delhi. Ms. Anjali is working since last 30 years as clinical dietitian in the prestigious institute and presently working as with Maharaja Agrasen Hospital. She had worked in MP Heart Center Delhi and Umkal Hospital from 95 to 2000, Orthonova Hospital from 94 to 95, and Bhupinder Nursing Home from 93 to December 93. She's a life member to ITA Nutrition Society of India and IAPEN. We welcome you, Ms. Anjali, and over to the chairpersons for the start of next session.
Dr. Bhavna is first.
Good afternoon, everyone. We have a great session. Previously, we learned a lot from the previous session. Now we move to the next session after lunch. I am privileged to welcome Dr. Bhavna Gandhi. She is Chief Dietitian in Lok Priya Hospital, Meerut. She is PhD certified diabetic educator, trained bariatric dietitian, trained keto dietitian, naturopath, yoga master, pranic healer, and she is associated with many other societies. Former executive member of IDA Delhi chapter, treasurer of IAPN chapter, Meerut chapter, Association of Diabetic Education, Nestle Nutrition Institute. She got many awards like Shield of Honor from Boston Heart Foundation, Meerut Ratna from Government Funded Institution 2013, Award for Health Icon of Meerut from Honorable MP Shri Rajendra Agarwal ji in 2016. She got Shield of Honor from Uttar Pradesh Government, Meerut of Women Empowerment 21. I welcome Bhavna on the Bhavna. To start with.
A very warm good afternoon to all. As Shilpa said, coming after lunch is quite difficult. and it's difficult to be awake also, I know that. First of all, thank you very much for your kind words, and a big thanks to IAPN Delhi chapter for giving me this opportunities. It's really a privilege to stand in front of esteemed audience and your mentors and teachers and giving a talk. Nevertheless, all of us are aware pre and probiotics for human consumption is still in infancy stage. But yes, we are going to see the significant use of pre and probiotics in the medical as well as nutrition. And we do hope so that they may be used further for the prevention as well as the treatment. Since morning, we had a good knowledge feast, good knowledge about prebiotic, probiotic, what are prebiotic, probiotics, the use of pre and probiotics in the brain, gut-brain axis, kidney, a lot have been covered and a very knowledgeable feast. My topic is emerging trends in pre and probiotics in hospital nutrition. The moment we say hospital nutrition means hospital. It's not a good name to be heard. A patient who cannot be taken care at OPD are generally the critical patients who are being shifted to the hospital. And with hospital nutrition, the theme of IAPN today is the medical nutrition as well as in the malnutrition. So hospital means a patient have been traumatized physically, that means diseases there, psychologically, a trauma coming to a hospital, emotional trauma, a patient have been under the stress. And malnutrition is very common. It have been very well depicted already in the IAPN ka video कि how come the malnutrition peeps in when at the time of the admission it is just the 31% of the patients who are severely malnourished, and by the more than 15 days or equal to 15 days it comes out to be 79%. And when at the time of admission it is 60% who really need the management, by end of day 15 or so it is 101% rather who needs a dietary management and interventions and supplements. And as very well depicted, malnutrition frequently are the underdiagnosed and the untreated among the hospital patients. There have been a data which is already depicting. If we see in the second graph, there is increase in the malnutrition frequency with increase in the length of the stay, with increase in the number of the patients also. So malnourished hospitalized patients leads to poor outcome. Why? Because 3.5 higher in hospital deaths are being because of malnutrition, 1.9 times longer stay, 2 times higher discharge rate to long-term care or rehabilitation centers, 1.4 times higher need for home health care services. They are not up to their own, attendant cannot manage them, so they need a help, and they are high economic burden on society, on family, on everyone. So it's $58 billion total costs which have been associated, and hospital cost for patient with the malnutrition is 73% higher than those without the malnutrition, and readmission possibilities are more. So we can see that how come malnutrition is an important part of the hospital nutrition. So if we see the graph, increased metabolic rate, anorexia is there, acute phase response is there, negative energy balance is there. This is all because of the infection which is leading to the malnutrition, and malnutrition is growing because compromised barrier defenses, impaired cellular immunity, impaired humoral immunity, and increase in the severity. So this is a vicious cycle which is going on, which says that undernutrition is there, and therefore loss of immunity is there, loss of immunity is there, increase in risk of diseases, infectious diseases. So again there is increased energy requirement, but that is not being able to meet. Despite of all our facts and all our 100% efforts, we are still not being able to meet. The reason is being again the trauma, the stress, everything, and the most important reason is the malabsorption. Malabsorption is very common. It is a state arising from abnormality in absorption of food nutrients across the GI tract. Impairment can be of single or multiple nutrients. If we see, this is the gut, and nutrients are no longer effectively being absorbed in the small intestine, whether that is the macronutrient or the micronutrient. This is all there because of the malabsorption, and because of the malabsorption, this malnutrition is there, and this is the vicious cycle what we are seeing. So if we see, since morning we have seen the gut-brain axis, the vagus nerve, the stomach, small intestine, large intestine, and we have learned what is prebiotic, probiotic, everything. So I'm shifting my gear now from here to the pathogenesis of these microbiomes which are there. Symbiotics are prebiotic plus probiotic. They have already been explained, and the benefits of having them is there, that it lowers the cholesterol, antibiotic associated diarrhea, lowers blood pressure, so many benefits. So if we come to this prebiotic, probiotic, postbiotics, symbiotics, they are anti-pathogenic, anti-obesity, anti-diabetic, anti-allergic, anti-cancer. So many benefits have been pointed out. Now, the prebiotics and probiotics have been put in the regulations which have been passed by FSSAI in June 2022. These are the regulations which have permitted prebiotic as well as the probiotic. But minimum viable number of added probiotic organism in food in the recommended serving shall be not more than in less 10 raised to power 8 CFU size per day. Still very not clear regulations, but yes, we can point them. And these are the list of the probiotic microorganisms which have been given permission by FSSAI. They are generally lactobacillus, bacillus, bifidobacterium, and the different strains of these. And regarding the safety norms, the regulations, we have been using prebiotic and probiotics. If we talk about kanji, if we talk about matha, if we talk about achar, shalgam achar, we have been using them since long. We can say that elders were good scientists enough. They have been using the prebiotics. We have been using them traditionally. But yes, since morning we have been discussing, no cut recommendations have been yet given. Kitna, konsa, kaisa. That still needs to be worked upon. And no general minimum level for probiotic is there. It has to be strain specific. As we are coming to the MNT, the individualized, tailor-cut, personalized nutrition, and so is with the probiotics also. This is the research which is in favor of emerging trend probiotics for human health. These are the 6 strains generally which are being used: Bifidobacterium, Lactobacillus, Streptococcus, Bulgaricus, Propionibacterium, and Lactococcus. So the benefits of few strains is this, that Bifidobacterium helps in obesity, Lactobacillus in oral health, E. coli helps in osteoporosis, Bacillus in oxidative stress, because we say that stress is also a main reason. And Lactobacillus acidophilus works more for the regeneration of the skin. Salmonella thermophilus is more for the nutritional status, Saccharomyces for immunity, and Lactobacillus rhamnosus is for the intestinal health. So now I take you to some stories of the researches which are there for being used in the critical care. The very first is the central role of gut in the intensive care. Critically ill patients undergo early improvement of their gut microbiota due to routine antibiotic therapies and other environmental factors, leading to intestinal dysbiosis. In the morning, we have already discussed. So there is a shift from microbiota to the pathobiota. That means there is the invasion of the pathogens are there. The key players in this communication is the gut microbiota. So if we see, as being talked in the morning also, the different access which have been made by the gut microbiota to the different organs. GBA, gut brain axis, already been discussed in detail. Kidney and gut have been discussed in detail. Gut and lungs will be discussed by the next eminent speaker. So if we see, the gut microbiota is the center which have the link and the relation with all the other major organs. So harnessing the gut microbiota in the ICU improve the management of several challenges. Targeting intestinal bacteria has the potential to preserve the restore barrier integrity. So current clinical strategies and using of the adjuvant treatment for dysbiosis in ICU. So that is the nutrition pro symbiotic fecal microbiota transplantation. Very new, but yes, we are working. So placing the intestine at the center of critically ill patient may play as a jack of all trades cards. The next study is on the acute diarrhea, which have been done and which says that probiotics significantly reduces the antibiotic associated diarrhea by 52%, reduce the risk of traveler's diarrhea by 8%, and that of acute diarrhea of divers by 34%. The protective effect did not vary significantly. So again, the effect of acute diarrhea is dependent on the age of the host and the genre of the strain being used. The next study is on the Lactobacillus casei shirota, which reduces this has been done with the commercial probiotic product, which reduces the antibiotic associated diarrhea in patients with the spinal cord injuries. So antibiotic associated diarrhea is a common complication of the antibiotic being used. The frequency of antibiotic associated diarrhea can be prevented as high by 60% during the hospital outbreaks. So based on the evidence, it was being found that significant decrease in the incidence of antibiotic diarrhea, associated diarrhea in patients with spinal cord injury. LCS is considered as a valid measure for prevention of AAD in the patients with spinal cord injury. The next study is the probiotics, prebiotics, and symbiotics in the chronic constipation. In many diseases, and evidence have been done, many researches have been done. There are so many still to be done. So it has been found that in human trials, inulin emerged as a promising prebiotic. With the probiotics, B. lactis, L. caseae shirota were associated with increased defecation frequency, with the latest strain demonstrating greater efficacy in improving the stool consistency and constipation system. They decrease the colonial transit time. So that is why they are much being helpful in reducing the constipation. And the probiotic effect on constipation symptoms may be the strain dependent. The next study is on the cardiovascular diseases. We have been seeing so many CCU patients, and cardiovascular diseases have been the leading cause of death in the developing countries in the last 15 years. By 2030, deaths will exceed 20 million per year. So we can very well see the data. It is going to become like an epidemic. Evidence from research suggests that there is a strong correlation between the gut microbiome and the development of cardiovascular diseases, because they help in digesting the probiotic go and digest the bile acid, then for that bile acid they need to use the cholesterol. So in this way it's just a rough thing. What I'm trying to tell you in brief, the detail is very much there over here, the chemical thing. The probiotics and prebiotics may help treat the metabolic disorders. It has already been said and shown in the morning, such as obesity, diabetes, cardiovascular disease. They have been shown to defend against cardiovascular diseases by lowering the cholesterol levels, cutting down on oxidative stress, balancing the functional and structural alterations of gut microbiota. The role of probiotics in cancer prevention have already been seen with the daycare OPD centers, with the chemotherapy usages, with mucositis. So an imbalance in the gut microbiome can lead to pro-inflammatory immune responses and the initiation of the disease processes, including the cancer. The main advantage of having them is that ensuring proper balance between the bacteria that are necessary for normal function of the organism and pathogens. These are the type of cancers: colorectal carcinoma, bifidobacterium, lactobacillus. In liver cancer, lactobacillus is being used. In gastric cancer, lactobacillus. So the different type of cancer and the different type of strains. None of one thing, one strain, one disease, this strain. It is still need to be worked upon. So the effectiveness of probiotics only for potential prevention of cancer or as adjuvant treatment during the anti-cancer chemotherapy have been suggested. The use of probiotics for post-operative patients in cancer surgery, if we see this probiotic, it leads to the increase in beneficial bacteria, which leads to metabolic activity of the intestinal microbiota, decrease in pathogen bacteria, decrease in carcinogenic compounds. So that is why in modification of the intestinal microbiota have been found useful to improve the capacity of the gut ecosystem to survive from surgically induced injury, resulting in fewer post-operative infections, thus maintaining gut microbiota balance and diversity is important for enhancing the host defense, especially during the recovery from a major surgery. We have already seen the catheter is rare, especially in the female patients, and bacterial vaginocytosis is very common in the patients. So the use of pro and prebiotics have been seen in the treatment of the bacterial vaginocytosis, which is very common infection for the vagina, and these are the different strains of lactobacillus which have been seen working for that. So in this study, it was being done by pre-pro alone also was being done, and it was being done in association with the antibiotics also. So in both the studies, the beneficial effect of pre and probiotics have been seen. So the probiotics may have a positive effect on the treatment of female bacterial vaginocytosis, alter the composition of vaginal microbiota, and prevent intravaginal symptoms. It is not just for the hospitalized patient, the beneficial effects have been seen for the postmenopausal females also and for the pregnant females also. So the prebiotics as a tool for the prevention and treatment of obesity and diabetes, very common. India is a diabetic capital, and we know that all of them are being associated. So in obese and diabetes, the insulin, there is increase in the insulin sensitivity. So with the use of prebiotics in the diet, there is increase in the insulin sensitivity. So the gut microbiota influences host metabolic disorders via the modulation of metabolites. So an overview in the improvement in the health of obese or diabetic patients treated by modulating their gut microbiota using prebiotic supplements in a regular diet. Like we all have been doing that. We have been telling our patients to use the lot of salads, right? Administration of prebiotics has the potential to modulate GM composition in patients suffering from diabetes and obesity and can be used as a therapeutic approach to cure the adverse effect of metabolic diseases. So the daily intake of prebiotic in a designed diet, so again it is an individual diet, has a major influence on gut microbiota by decreasing the gut permeability and bacterial translocation and reducing LPS induced inflammation. So if we see, there is a significant evidence in the reduction of diabetes and obesity. Oral health is very common in the hospitalized patients, which is again being ignored. Generally ये वाले parts रह जाते हैं. So oral health has also, there is a benefit of use of lactase HNO19 in oral diseases, especially if we see the mucositis, the cancer patients. So the beneficial effect have been seen. This is the healthy gum and teeth, and this is the inflammation. So there have been the benefits. The oral cavity is one of the environment of the human body with the highest concentration of microorganisms that coexist harmoniously and maintain homeostasis related to oral health. The study says that the use of the lactase HNO19 have shown the benefit to the host as it decreases the pH in the oral cavity, so that the plaque bacteria cannot form the dental and the calculus that cause periodontal diseases. The gut microbiota and immune system, as it have been seen, it has to be given with a lot of care. We cannot give a probiotic to immunocompromised, but yes, there have been studies. This graph have already been explained in the morning, that how they good prebiotic give food to the probiotic compete with the colonization and nutrients, and they may inhibit the growth, and then modulating the immune response, improving the gut barrier integrity, and providing the energy also. So they are maintaining the microbiota. The effect of this is a very short term studies. which have shown the use of Lactobacillus cesae shirota in ingestion to a safe, supportive approach with immunological and biological benefits to the HIV infected children. But this was a very small study. So again, a lot of work needs to be done. And the microbial therapeutics in neurocognitive and psychiatric disorders. We have already discussed in the morning the gut-brain axis. So this is the gut microbiota, Lactobacillus, Bifidobacterium, Saccharomyces. So these all help in releasing the neurotransmitters which help in the good health of the brain, the central nervous system. So the effect of these probiotics have been seen much more. The role of these are being observed and the studies are being conducted to see the effect in Alzheimer, depression, anorexia nervosa. And if we see, this is the microbiome technology for Parkinson's. There are studies which are in favor of using post and pro and prebiotics in the treatment of the Parkinson's disease. A reduction in tremors have been seen. So microbial therapeutics are a new psychotherapeutic approach. Effect of symbiotics, probiotics and prebiotics in NAFLD. So NAFLD is a chronic fatty liver disease found approximately in 25% of the population. So the incidence is varying from 19 to 86 per thousand. And the benefits of use of symbiotics, probiotics are likely to be the most potential effective treatment in the for AST, ALT reduction in adult patients with NAFLD respectively. This is a very latest research which is being done. So again, we need to go for more randomized controlled trials for this. The next is the effect of probiotics in the low FODMAP. Now we know everyday FODMAP diet, it is becoming, IVS is becoming very common. So in these cases, when the study was being done with the low FODMAP diet versus prebiotic supplementation, by 4-week administration, both treatment has opposite on intestinal microbiota, but some clinical benefits were being seen. After treatment discontinuation, the decrease in symptom persisted in the prebiotic group and they disappeared in the low FODMAP diet. So it is being suggested that intermittent prebiotic administration might therefore be an alternative to dietary restrictions for patient and functional gut symptom. The same study have been done on the role of FODMAP diet and probiotics on gut microbiome in IBS. So in this, again, a positive correlation appears to exist between the low-fat FODMAP diet and IBS symptoms. The probiotics are taken amongst IBS patients. A reduction in symptom is also observed. Because if a patient is on low FODMAP diet, the moment you stop up with the low FODMAP diet, there is decrease in the bifidobacterium, which is a positive bacteria for the human health. But if they are being given with the probiotics, then there is increase in the abundance of bifidobacterium as well as the lactobacilli. Impact of prebiotics and probiotics on skin health is also being seen because we see the bed sores also. So the study was being done applying them tropically, but more benefit was being seen when they were being ingested. So nutritional products containing prebiotics and or probiotics have a positive effect on skin by modulating the immune system and by providing the therapeutic benefits for atopic diseases also. The researches are being done more in pediatrics and more beneficial results are there, but still because of paucity of data, it still needs to be done a lot. I cannot promise that. So this says, now as we have seen the effect of postbiotic in all the diseases, now it is being there in the plant-based proteins also. All the new products which are coming, they are being added with the probiotics. In pediatric, they have already been added, but in adult new products, the high protein mixtures are coming with probiotics. And the result of the study was that the concentration of branched chain amino acids and essential amino acids were 1.36 and 1.41 times higher in the co-administered group. So the co-supplementation of plant-based protein with lactobacillus casei can be suggested to improve protein digestibility. So we at Lokpriya conducted a study, just 8-week study, informal study on the use of the scientific from commercial probiotics, and the patients did found relief in various digestive issues. 91.9 experiencing relief in flatulence and in constipation. The probiotic also demonstrated appetite stimulating effects, as 82.5 reported increased appetite and 80% felt hunger pangs after consumption also. So our study has provided a valuable insight into the experience of individuals who have consumed probiotics. The responses indicate a positive overall perception of a probiotic effects on various aspects of health and well-being. So furthermore, probiotic appears to positively impact immunity with 83% immunity. And we want to say this, that probiotic is well received and may offer various digestive and appetite related benefits without significant side effects for a majority of those who were surveyed. However, it's important to keep individual health in consideration in mind and consult with the healthcare professionals when making our dietary choices. So the trends in the hospital nutrition, what we are using is a use of xylem. We have been seeing our patients isabgol ki bhusi khate hue bade time se suna hai. So this is the way what even we are using, because our hospital is catering to our peripheral patients where affordability is a very big issue. So to them, what we try, to the constipation patients, we give them 10 grams per day in the milk at night. And in case of diarrhea patients, we give 5 to 10 grams in curd during the daytime. So the research is there in favor of that using the xylem. The reason is being that xylem supplementation increased the stool water, and this was associated with significant changes in the microbiota and most marked in the constipated patients. The second is the use of the fenugreek. We soak 5 grams of fenugreek in glass of water. This is not that just we do, it is a teamwork. It is with the due consultation of the consultant also. It is like we go in the teamwork, and then we soak the water, methi dana in water, give the water in the morning, and if a patient can chew, we tell them to chew. If they can't, then we do provide the powder, which can be used in the curd or dal or dahi while consuming it. So the galactomannan from fenugreek seed showed prebiotic potential and was found to serve as a nutritional source as well as carbon source for enhancing the growth of probiotic strain B coagulans MTCC 5856. So it is an ideal symbiotic which can be used. The probiotics which are being used in the hospital nutritions are pickles. Yes, with the low salt consideration we do provide, but that depends again from patient to patient. Kimchi in our corporate hospitals, it can be given. Then the yogurt, Greek yogurt, probiotic yogurt, if the patient affordability is there, yes, we do go ahead with that. Buttermilk is always the grandma probiotic. We being in UP, हमारे यहाँ तो मट्ठा चलता ही चलता है. Matha is like pani. You have to drink it. Then curd, most common probiotics. Paneer is an Indian cottage cheese. बहुत खिलाया जाता है बचपन से. Then cheese, lot of cheese are there. Idli, fermented source. Yes, it is a part of our hospital routine kitchen. The dairy products which are being known as the desirable delivery vehicle. These are the prebiotics. Bananas, a versatile budget-friendly source of prebiotic to be delivered at 11 a.m. as a healthy snack. Apple, garlic, yes, if there is no religious belief, then we do use that. Kiwi fruit, again, if the patient can afford it. Legumes, it's a part of our regular menu. Oats are being used. Potatoes, if a patient health permits, then broccoli, radish as a form of salad. Flax seeds, yes, if a patient can afford flax seeds, if required, we do provide, otherwise we do tell the patients. And apple, again, a mid-morning healthy snack. So the conclusion is this, that the intestinal microbiota forms a diverse and complex ecosystem. However, there is much variability in bacterial numbers and populations between the stomach, small intestine and colon. In comparison with the other regions of the GI tract, the human colon is an extremely densely populated microbiota ecosystem. These gastrointestinal microflora are important elements in the health of the host animal. Environmental factors like diet, medication, stress, physical, psychological, emotional, can all adversely affect the composition and or activity of the gut flora. The deficiencies created can be repaired either by added viable organisms, that the use of probiotic, or by stimulating specific components, like example is bifidobacterium of the flora with some chemical or the plant type prebiotics. Pre and probiotics have a long history of usage, especially in Indian history. We have seen and are increasingly becoming better characterized and widespread in dietary supplements. While many factors from genetics to environment, particularly diet, influence the intestinal microbiome, the efficacy of probiotics have been demonstrated not for intestinal microbiome changes but for specifically clinical outcomes. So, lot need to be done. Authorities such as ISAPP, CRN, IPA, WHO, FAO have established guidelines for probiotics and organizations are working on it. Recommendations are being given to probiotics but not freely available. So, while continued research of pre and probiotics and the human microbiome is necessary in healthy and diseased populations to investigate the potential diagnostic and therapeutic solutions. So, we can say that pre and probiotics are smart clinical tool to promote patient health in a variety of conditions. Thank you very much for your time and attention. Thank you. In case of any queries.
Excellent session.
Thank you, ma'am.
I would say this is a bullet.
Thank you
One
very much,
bullet
ma'am.
for all.
I'm overwhelmed that listening it from your teacher is really a
It's
gratitude.
amazing. Did you leave any disease out?
Pediatric छोड़ दिया, वो मैम करेंगी. क्योंकि मैम is there, the master is there. I left pediatric. I just touched it because ma'am will cover it.
But you've taken each disease and explained it so well.
Thank
We
you
were
very
so
much.
scared using it in hospital scenario earlier. But you know, when we hear lectures like this, and all supported by science, all supported by studies, which you just feel now you're armed with knowledge and information and you can just use it without feeling scared. Will something happen to that patient? Will the doctor catch your neck? Did you give probiotics? This happened post. It happened initially, you know, if something happened, you're not supposed to give it in ICU. But you've done it very well and you've taken each disease separately. So I think the answer is we just had our shot in the morning. We just take it every day. Maybe the medical doctors will soon be out of business with the probiotics.
Thank you very
So
much.
questions open. Any questions from the participants?
I believe I've
Very well.
over-explained.
Explained it very well.
Thank
एक टेबल बना दे यार. इतने
हाँ.
सारी चीजें बोलियो कैसे याद.
Obesity, diabetes.
Ma'am, I have tried to
Little
cover.
skin.
Actually, I was being given the hospital nutrition, and hospital means everything. It was such a big basket for me to be catered.
Maybe you know what is the references, what is the sample size, and this you need to see all.
Ma'am, I have already given the links niche.
हाँ,
It
but
was difficult for me to cover it, so I've already given every link over there on each and every slide
Ma'am, this is
below.
present on the YouTube link. So आप वहाँ से pick कर सकते हैं and you can
The every
use
link
it.
of every research is being there.
including dental, including skin.
Everything, ma'am. I tried to cover each and everything.
Because I have seen so many patients, मतलब like in my family only, कि उनको इतने टाइम से फीवर था और वो रूल आउट नहीं हो रहा था. आई जस्ट टोल्ड देम कि आपने एक बार डेंटल चेकअप करा लो, और वो डेंटल चेकअप के बाद उनका रूल आउट हो गया, वो तभी वो ठीक.
It is there, ma'am. It is there. It
The
is there.
mouth, including UTI, having catheters. Beautifully done. Excellent.
Thank you very much, ma'am. Thanks a lot. Thank you.
Yeah, but we used this isabgol husk through her childhood. Today we know the science behind it. And you always used it like this, dahi for diarrhea, milk for
Ma'am, to add it,
water for in summers.
and ma'am, to add it, we are using coconut oil also. With
For
milk, with xylem, the isabgol, in milk, with 1 teaspoon of coconut oil. Sir asked a question in the morning, sir, it is very new research, but a research is coming up that if you apply the coconut oil at the anal region, that may help in increase in the microbiomes over that area, which may help.
It is still in very infancy. Not exactly. So इसलिए I couldn't put it up.
I think the best thing would be there's a lot of traditional wisdom in India,
Yeah, ma'am.
China, Japan. Combining it with the modern science.
Ma'am, we are going back
That
to
is the
Bhale
way
wala
to
thing.
go forward. I know. So I think that is a way to do.
Post-COVID, all of us are awakened now that our elders were scientists. They knew everything before and what we are knowing now.
Thank you so much, Bhavna. Actually, Bhavna was the only speaker who has taken the topic from me. Rest all have decided on their own. So I thought, let it be from the hospital view point when we have been practicing into the hospital. Thank you for doing justice to the topic. Thank you. So I'll request Ms. Anjali to present a token of thanks to Bhavna.
इधर 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 अच्छा
Thank you. So I didn't find anybody sleeping, just shows that I was yeah, everybody was awake. We'd like to invite Dr. Mahesh Ravi Kumar Jinsari for the next session. So we've heard so much about CKD, gut. Now we're going to hear wonders in pulmonary. He's HOD Department of Pulmonary Medicine, AIIMS Kalyani, Kolkata. He's postgraduate from Mumbai, now associate professor and head in pulmonary medicine, AIIMS Kalyani. Over to you. So another aspect to learn about. I think we should ask Yakul to bring up their productions. By the time we go home, I think we'll all be using more and more of this. Over to you.
So, very good afternoon. It is a very tough job for me because the speaker before me, Dr. Bhavna, has given such an excellent talk. So, I don't know whether I would be able to give justice to my topic the way she has done. So, at the end of my session, we'll come to know whether you have understood everything or you have slept. So, till now, she has managed to keep all of you awake.
Adjust कर दो, right? Yeah.
So, actually, about this prebiotics, probiotics, symbiotics, and gut microbiome, you know things better than me. When I was given this topic about the gut health and lung, or simply we can say that lung gut axis, this is one of the very neglected axis among all the axes. Because as you see that Bhavna madam has given one chart where the symbiotic and the a bullet like a clockwise manner was there, and I was just reading through each and every bullet and I was trying to find is there a mention of lung. So there was no mention of lung even there also. So can you imagine the negligence related to this gut lung axis? So
Sir, I mentioned it. I said
no.
She left it to you.
Yes.
I left it for you to speak.
No, ma'am, you mentioned it, but it wasn't there in your bullet. That's what I'm trying to say. Okay. So, as all of you know that the human cells versus the bacterial cells, the body has around 30 trillion human cells, and more than a human cell, we have a bacterial cell. That is 40 trillion. And when I googled about the trillion, the number of zeros I cannot remember. That's why I put it there. And bacterial cells outnumber the total human cells by a factor of 10, and the genetic material coded by the gut bacteria. Do you have an idea how much genetic material coded by bacteria as compared to human cells? Just any rough guess? It's more than 100 times what we have. So our body is regulated by our microbiome, but we focus mainly on our human cells rather than focusing on the microbiome. So these are important terms. All of you know these terms, but it is very important because it always confuses me what is microbiota, what is microbiome. So I remember it by if it is a T is there, that is a type of microorganisms. If there is a M, that is microbiome, that is a multiple genes from all the microorganisms. Then prebiotic is the enteric food for the host benefit. Probiotic is the living organisms for the host benefit. Eubiosis is a normal flora. Dysbiosis is an abnormal flora, and symbiosis is when both of them are benefited. But when we mention as a commensal means one is benefited but the other is unaffected. If it is in pathogenesis, then one is benefited, other is affected. So, as I mentioned earlier, lung microbiome is extremely neglected among all the axes. In 2007, NIH is a US body. They have conducted a big trial as a human microbiome project. And that time they did not give much importance to lung microbiome, thinking that lung is a sterile organ. The culture methods available, they couldn't grow many things, but then as we are having a modern techniques like molecular diagnostics, we know that lung is no more a sterile organ. So human microbiome is a microorganisms that live in and on the human body. They have been living with us for thousands of years with a symbiotic association, and that's an home to from bacteria to viruses, etc. And they have multiple roles like immune regulation, immune modulation, digestion of food particles. As a byproduct, they produce vitamins. Those are very helpful to us, and they have even a role in prevention of diseases. So this figure shows that there are n number of bacteria of normal human microbiota. The names are like actinobacteria, Firmicutes, Proteobacteria, etc. And different body parts is having a different predominant microorganism. Like we can see oral cavity, Firmicutes, Proteobacteria in stomach, then Firmicutes again in vagina, and again Firmicutes in colon. So this has been found that those people, children, those who are born of normal vaginal delivery, they are exposed more to Firmicutes from the vagina, and their gut had more predominant component of this Firmicutes, which is a normal component of colon, which you can see here. And those children, those who have a normal vaginal delivery and high content of Firmicutes, they had a lesser chances of having asthma or atopic disorders in their lifetime. So normal lung microbiome, the healthy lung is having a low microbial density, that is a less number but more diversity, as compared to a lung disease where they have a high density of microorganism but very low diversity. And as all of you know that it's a strict anaerobes are there in gut, and more prevalent taxa are these three names that are very difficult for me to pronounce, very difficult to remember, like taxa and genera, everything. So I always remember that bacteria is a foreign body to humans. So I remember by remembering it as a foreign body, RSVP. RSVP is respond if you please. That is always written on a, what we say on this invitation cards. So it's the most prevalent taxa are Firmicutes and Bacteroides, and the genera are Streptococcus, Veillonella, and Prevotella. So this has really this has given me little relief that at least I can remember them in future. Otherwise, these things are very difficult to remember. So normal lung microbiome, as I mentioned that earlier, we had a misconception it's a sterile organ, but that's no more a truth. It's a dirty organ having a multiple microorganisms. It is having very diverse number of microbes, as well as the number of these microbes is not high number. It is a scanty number. That is a decreased number. Why there is a decreased number of microbes in lungs as compared to gut? Because the primary function of lung is the respiration. If that part of lung is inhabited by a lot of microorganisms, it will interfere with a primary function of lung, that is respiration. So that's why number is low. The colony over there are very dynamic and very transient. They keep on changing with time, and this change is determined by how much organisms are coming to lungs and how's your innate immune system and how is your clearance. And most of these are derived from oropharynx and gut. So again, we need not remember the whole of the chart. Only thing which you have to remember, the upper respiratory tract is having high biomass and lower respiratory tract is having a low biomass consisting mostly of oral commensal. If you can see that this dotted line is a difference between this differences between the upper respiratory tract and lower respiratory tract. So upper respiratory tract, it's above the vocal cord, lower respiratory tract is below the vocal cord. And the mechanisms by which these organisms reach lung are oropharyngeal aspiration and GID, there is a GERD, as well as microaspirations. And the clearing mechanisms are like cough, mucociliary transport, and innate immunity. So we have to maintain a balance between these two. So this balance is maintained by the microbial immigration as well as microbial elimination. So there are certain factors which favor the eubiosis and there are certain factors which favors the dysbiosis. So what are the factors those favor the eubiosis? These are like breastfeeding, normal vaginal delivery, then healthy diet and exercise. On the contrary, those who are fed with formula feed, those who are have unhealthy diet or exposed to a lot of antibiotics, or those who are have obesity, they have a more tendency that they can have a dysbiosis. And this balance is maintained by all other factors like what is your age, what is your exposure, exercise, diet, your genetic makeup, as well as your immunity. So how lung and gut microbiota are different? As I mentioned earlier, lung is having low amount of bacteria as compared to gut. It's a transient dynamic. However, for the gut, it's a locally replicating and it is a resident of a gut. For the lung, it is derived from the gut. Gut, it derives from since we are born, it is derived from the environment or from the whatever things which you eat. Lung microbiota mostly influences the lung, and some amount of the effect is seen on the gut. However, the gut is actually also mentioned as an enteric nervous system. That this nervous system doesn't even control the brain, but it also controls the lung as well as immunity as well. So if we say that this microbiota is an ecosystem, then we can say that lung is a desert and intestine and gut is a rainforest like an Amazon. So these are the different axes as I mentioned earlier. So gut lung axis, we can see that these two parts, they share a continuity with a normal anatomy, that from the oropharynx it divides into two. One goes to lung and one goes to intestine. So they have a common anatomical continuity, and they are in sync with each other. They communicate with each other, and they communicate with each other via blood, via lymph, and via immune cells. So as you know that lung is the only organ in the body who is constantly exposed to external microbes. So once these microbes reach the pulmonary system, these microbes are encountered by a innate system, and then your genetics and load and virulence of this decides that what will be the outcome of this. And as you know, some of the microbes which are inhaled, they are trapped by mucus, they are trapped by the nasal hairs, they are trapped, they're removed by cough reflex. And this mucus or trapped microorganisms, all of them are not coughed out. Most of them are swallowed also. So these antigens, when they are swallowed, they are processed by a gut microbiome. This is presented to immune cells, and there is a immune regulation related to it. So we'll see how that immune regulation goes. And in this second diagram, we can see here that there is a constant efflux of lymphocytes from lungs to liver and from liver to lungs. As madam has mentioned in the previous lecture, when we have critical care patients, in critical care patient, there is a chances that there is a translocation of gut bacteria to lungs or to other parts of the body, and when there is a translocation, it favors the poor prognosis. So here, when antigen is inhaled, it is exposed to innate immunity, and this innate immunity presents the antigen to lymphoid tissues like lymph node and spleen, and this is a part of adaptive immunity. So body will make antigen specific T cells, and this antigen specific T cells, when they are encountered to a same antigen future, we will have a faster reaction to the such kind of antigens. Same antigens, when they are swallowed, they reach gut, it is processed by the gut microbiota, and gut microbiota, this antigens are body learns from this antigen that whether this antigen is antigenic, it is infectious, or it is a low infectious. So it decides between, there are two kinds of immunity, Th1 immunity, Th2 immunity. Th1 immunity is the immunity what we already know that when the pathogen comes to your body, body tries to kill it. But Th2 immunity is that immunity when you are encountered to such antigen, body will try to run on an allergic course. So that allergic course is Th2 immunity, and this gut processing of this antigen gives body some T regulatory cells, some Th17 cells, and these cells, this is a very complex ununderstood mechanism that body is trying to keep a immune reaction in control. What did we see in COVID-19? Some of us had COVID-19, we were asymptomatic. Some of us were symptomatic, but we recovered. And some of us, despite having a young age, no comorbidities, they had immune dysregulation and they passed away. So what did decide about this outcome of this COVID is your immune dysregulation. That whenever we are exposed to some amount of antigen, body shows a dysregulated immune reaction where the body will kill the pathogen, but at the cost of killing the own body cells as well. So this immune dysregulation has led to a mortality in COVID that we already know. And this processing of this gut bacteria, they have very important role in a controlled immune reaction. That is why gut is very important. And apart from this, some of the thing that one thing which we always forget that uh, as we know, every smoker will not develop a COPD or every smoker will not develop a malignancy. Or there are some people, even though they are non-smoker, they are exposed to some amount of passive smoke, they will develop a cancer. So the one of the very important factor behind each and everything is your genetics. So we must never forget about our genetics. So dysbiosis, as I have already mentioned, this is just a deviation from our normal microbial composition. So these are little bit difficult, I would say very complex and very concentrated slides. So I'll just simplify for you. So in a eubiosis, some of the antigens are aspirated and these antigens are cleared from the body by immune reactions. And once this immune reaction happens, it is cleared and transiently there is an increase in a tone of immunity. Body increases the tone of immunity, control the antigen and goes back to normal. So this is a regulated immune check. However, when there is a dysbiosis, there is a recurrent aspiration and this causes some irreversible changes, and this is an early dysbiosis and it is due to the immune disbalance. So when there are recurrent aspirations, body is exposed to recurrent pathogen, then a stage will appear when the body will go into a stage of immune exhaustion. And when there is a stage of immune exhaustion, persistently there is an exposure of microbial organisms, a state will come that is known as colonization by formation of the biofilms. There are multiple chemical reactions involved in it, but the crux is that it is related to immune exhaustion. So as we know from here, COPD, can you see here, FeV1 is decreasing when the gold class is increasing and symptoms are increasing. So as there is a dysbiosis increases, the disease progresses. In second, this side we can see IPF. IPF is a form of interstitial lung disease where studies have shown that patients with high bacterial load had a poor probability of survival. When mice, those who had a bleomycin induced pulmonary fibrosis, when mice were germ free, there's a better survival as compared to a conventional mice, they had a worse survival. Again, in ARDS patient, when there is an increased gut associated bacteria, as I mentioned earlier, because of the translocation, it favors a poor prognosis. Even the lung cancer dysbiosis causes a higher stage, poor response to treatment, resistance to even immunotherapy, or patients even can have a pneumonitis. So dysbiosis doesn't even have effect on a one organ, but it ranges from all the respiratory diseases, right from COPD or airway disease to lung cancer. So this slide explains which I mentioned earlier about the Th1 immunity and Th2 immunity. Th1 immunity is a normal immunity, Th2 immunity is a allergic immunity. So this concept is known as a hygiene hypothesis. So what is hygiene hypothesis? If you remember your grandfather or your great grandfather, they used to live in a small house, simple house. Some of them used to coat their houses with the cow dung. Okay, they were not treated much often with antibiotics. They used to live in a big houses with multiple people. There's a joint family. So there was a constant, and they were very close to nature. There were lively stock like cows, buffalo, dogs, everything would be a part of their family. So they are constantly exposed to all kinds of microbial organisms. So their immunity was tuned that they didn't have time to switch towards the allergic pathway because they were constantly engaged or busy on a normal usual Th1 pathway. Then we became a modern, houses are clean, lot of antibiotic use is there. Even we have air filters that we are not exposed to air pathogens as well. We are exposed to a lot of vaccines, we are exposed to a lot of antibiotics. So what has happened, our body is not exposed to those many microbial pathogens. So what has happened then, our immunity axis has gone towards a Th2 pathway. So what is Th2 pathway will do? Even there is a small amount of antigen which is not that much harmful to our body, but body will place a very strong immune reaction towards that antigen, and the outcome of those strong reaction is asthma, allergy, eczema, etc., etc. So gut has a very important role in this immune dysregulation. So as we know that impact of gut biosis on health, it doesn't even affect the lung, but it's from cancer, mental health, liver disease to diabetes, everything and anything. So out of the 100% immune cells, gut holds a 70% of immune system, and early dysbiosis leads to a chronic inflammatory diseases. As this balance shifts from this symbiotics or these pathogens to pathogens, this dysbiosis will lead to chronic inflammation. So one of the theory behind is that this dysbiosis causes a leaky gut. So how does this leaky gut happen? As you can see, these are all microbiota on upper layer. This is a mucus layer, and there is a strong barrier against this microbiota is because of the mucus layer, followed by a physical barrier of this epithelial lining associated with tight junctions. So dysbiosis causes a loss of these tight junctions. Okay, this is a lipopolysaccharide, and these tight junctions are lost, and these immune cells will reach the circulatory system, and this will cause a chronic inflammation. So in a summary, how lung affects the gut and how gut affects the lung. So this table explains you COPD, what COPD is having effect on the lung and what effect it has on a gut. So in a nutshell, I can say that it adversely affects the lung as well as it adversely affects the gut. Again, similarly for a bronchial asthma, it's true. There is an increased risk of childhood asthma development when there is a gut problem early in life. Also in cystic fibrosis, as we know that cystic fibrosis, there are thick secretions are noted. This thick secretion causes a change in microenvironment around this microbiota, and this has a very deep and long lasting effects on immunity as well as those microbial organisms. So this explains the triad, how the lung health is affected by gut health, and this gut health is affecting the cardiovascular system. As all of us know that COVID was having effect on lungs, on gut, on your CNS system, on your immunity, on your blood coagulants, as well as it had effect on our heart. So whenever there is an acute bacterial infection, acute viral infection, it may cause myocarditis. Again, whenever there's a some of the people, they have viral infections, they can have a gut imbalance in the form of diarrhea, or if there's a dysbiosis of the gut, this causes an increased susceptibility of lung infections. So these are related to each other, where this explains that how this effector work with each other and communicate with each other. But the crux of story is each system affects the each system. So this shows that in a healthy state about angiotensin. Angiotensin is one of the very important factor for a body. It causes a vasoconstriction and it is having a pro-inflammatory action. So this angiotensin is cleaved by S2, and this makes angiotensin 1 to 7, and this angiotensin 1 to 7 is having vasodilatory effect and anti-inflammatory effect. So body will decide where we want a vasoconstriction, and body will decide where we want a vasodilatation. Body will decide where we want a pro-inflammation as well as anti-inflammation, and there is a very tight balance between these two things that is regulated by our immune system. So during the COVID, what happened? COVID used this ACE2 receptors to enter the body, and there was a dysregulation related to this ACE receptors, and because of this angiotensin wasn't converted into angiotensin 1 to 7. There was high amount of angiotensin 2, and this angiotensin 2 being a pro-inflammatory and vasoconstrictant, it has caused a lot of damages to the body. So again, weight metabolism, it has been seen that in lean patients there is a lot of bacteroides, and this produces the short chain fatty acids like acetate and butyrate. And as all of us know that butyrate being is having so many actions and so many good positive effects on the bodies compared to acetate. However, in obese patients there was a high number of firmicutes and methanogens, and this led to a increased production of short chain fatty acids. So some of the study, it has been found that even though patient has undergone some kind of bariatric surgery, there will be some patients they will have a weight loss, and there will be some patients they will not have weight loss, or they will not have a weight loss as much as other people. So the one of the theories behind it that they have an altered microbiome, and their microbiome was predominantly firmicutes, and this firmicutes was able to digest even the non-digestible products of intestine also. That is one of the theory that they had a weight gain despite having a bariatric surgery. So this was a study done on the mice where they have studied the low fiber diet and fiber rich diet, and the crux was there was inadequate immunity when they had a low fiber diet compared to a balanced immunity when they had a fiber rich diet. Again, this is the effect of prebiotic, probiotic. Already madam has explained everything, so I'm not going to go into details of this. So again, how does it happen at a molecular level? Controlling the immunity. So this high fiber inulin and probiotics, they produce a short chain fatty acid, and of which I would like to mention about the butyrate. Butyrate being having a very good effects on the body, and this causes the immune modulation at a bone marrow level. This butyrate and other products, they reach the bone marrow, and where they produce a AAM. What is AM? AM is alternatively activated macrophages. So these macrophages are not a usual macrophages. They are different kind of macrophages. They had fewer amount of the CXCL. This CXCL component is required to activate the neutrophils. So once this special macrophages reach the other parts of the body, including the lung, they will not produce a immune dysregulation or a very strong reaction towards any pathogen. They will have a controlled check on those immune reactions, and it will cause less neutrophilic infiltration to the lung as well as a moderate tissue damage. So this is one of the trial where in the trial they have checked that partly controlled bronchial asthma patients, they are randomized into two, placebo versus probiotic group, and they have seen that the 93% improvement in a probiotic group was noted. Again, there is this is a novel therapy which mentions that larazotide, it's an zolinil antagonist. This decreases the epithelial permeability. And once I have mentioned the leaky gut theory, so probably it may give us a protection from that leaky gut and decrease the chronic inflammation. As well as metformin, apart from having anti-diabetic effect, it decreases the weight, and it has multiple effects on the body, including on the tight junctions, inflammation, as well as alters the gut microbiome. When I was reading about aging, so in one of the aging, that how can we decrease the aging, there was a special mention of this metformin, because metformin is having a lot of effects in the body, including the inflammation, diabetes, as well as anti-aging. So these are the meta-analysis. This meta-analysis has showed that there is a probiotics is around 80% effective. Some of them have showed there is no positive effect. But the point which I want to deliver and convey to you is this has showed that there was none of the article has showed any negative effects. So this is a very common thing. A patient, specifically those who have lung infections, they keep themselves away from all the probiotics, natural probiotics, curd, buttermilk, etc., etc. They say that if I take a curd, I will fall sick, I will have cough, or I will have a mucus. So we have to mention that it hasn't showed any kind of negative effects. Again, this is during the pregnancy. This meta-analysis mentions that prebiotics and probiotics are safe even during and after the pregnancy and during the lactation. So this meta-analysis shows that it reduces the risk of atopic eczema in children. Around one of the systemic studies showed that there was no benefit, and some of them have showed benefit on at least one symptom in allergic rhinitis. So probiotics does have a important role or effect in allergies, as well as in the respiratory tract infection. There was substantial benefit noted in reducing the incidence and duration of upper respiratory tract in healthy people, and two of these analyses have demonstrated the positive impact on immune markers, one on the improvement in the effect of influenza vaccines. So do you have an idea about what is a project blueprint? Does anybody know about the project blueprint? Or does anyone know about the Brian Johnson? Have you heard about the Brian Johnson? So Brian Johnson was, he is one millionaire, he's a technocrat. He decided that he, at the age of 21, that I will earn money, and at the age of 30, I'll go for the retirement and I'll work on my health. But he admitted during the interviews that he got laid by 6 years. So he became a millionaire at the age of 36 years. And after that, he started working on his body, his nutrition. And he is claiming that as a part of this project blueprint, that he wants to, even though he's a 45 to 50 years old, he wants to decrease or reverse his biological age. So he claims so different things, and he has even shown some of the parameters with which, even though body has aged 45 to 50 years, but the parameters or the biological parameters of body are still young, 30 years, 35 years. This graph mentions everything. So one of the parameter, one of the thing on which he is working is gut, is nutrition, and is probiotics. So probably in coming future, we may have a better knowledge of this probiotic, and we can even know that whether this probiotic is having an effect like an anti-aging. Thank you. So I did not put a conclusion slide purposefully, because I know all of you know what is the importance of probiotic and prebiotic. If I was going to address a pulmonologist, I would have mentioned my conclusion slide as a, whenever you see any pulmonary patient with a chronic inflammation, refer the patient to nutritionist or dietitian so that they can take over further from here onwards. Okay, thank you.
Very nice conclusion, and I really thank you for that. And I want to ask you, you believe in not giving curds to asthmatic, because most of the time it's the doctors who tell the patients not to take curd, and then we try and convince them that nothing will happen. It's difficult. It's a battle. But Ayurveda supports it. So Ayurveda is also science. So what would your, and because of this probiotics or even taking Yakult again becomes into a curd base or a milk base. What is your take on that? For asthmatic.
I'll explain, madam, your question in a little different way. As madam has mentioned in one of the slides also, one of the probiotics, she uses a garlic, if it is a culturally acceptable. Okay. So there are some people, those who say, including my own mother, she follows Brahma Kumaris and she doesn't eat onion and garlic at my home. So it is such a strong belief. Whatever data, whatever science I'm going to present in front of her, it is not going to help. So if I give them something which is garlic, she will not eat it. So what is an alternative to give garlic or effects of garlic? There are some tablets available with a garlic extract. Okay. Sorry?
It's coming with apple cider also. The garlic with apple cider, garlic with apple.
Yes. So these are the things with which you have to deliver them. Now, our dadis, our nanis have told us that whenever you get sick, whenever you have upper respiratory infection, don't eat nimbu, don't eat citrus fruits, don't take milk, and don't take curd.
The medical team says
It is such a strong belief that we cannot just get away with this. Even the physicians also. And the one of the thing is that suppose we are exposed to a curd, which is a very over fermented curd. It's very khatta. So what will happen? It may cause with the sudden some pH changes in the throat, and it will cause some irritation. And this irritation is sometimes thought that my disease has increased. So it is always better them to have a very fresh curd, freshly prepared curd. If not, then the freshly prepared, or if we are not able to give them directly this thing, sometimes it becomes difficult to give them a diet, but it becomes very easy to give them a medicine. So give them a probiotic in the form of medicine rather than giving them a curd or this buttermilk.
Very well presented, Dr. Mesh. Absolute congratulations to you on this. And what do you say to the Delhi air quality and the probiotics? Does it help?
Madam,
no,
this
not
is
for the future, because all of us are facing it. And the winter's around the corner.
No, ma'am.
The burning in Punjab, all that. So, no, what does your experience say? Does it help? Really help?
So does air purifiers help? I don't believe in it.
The probiotics help in these people, and most of them are complaining of cough and irritating cough, which is nothing to do with infections.
No, ma'am, I do not have a data to conclusively say that the probiotic will give you a beneficial effect from your air pollution. But looking at my previous slides where I have mentioned that the gut is having a very important effect on your immunity. If you are able to make your gut very strong, then probably indirectly I can say that logically it will surely give you benefit from the air pollution as well. Because what will happen when we are exposed to such air pollutants or some smoke and everything, you will have a higher chances of getting a respiratory infection. And one of my earlier slides I have mentioned that if a probiotic can give you effect from upper respiratory tract infection. So surely if you are not going to go away from this environment, with probiotic you will have a 2 infections. Without probiotic you may have a 3 infections, but I do not have a data to support my hypothesis. One more thing I would like to clearly mention that a few years back I used to stay in Mumbai, heart of the Mumbai. The Mumbai climate is, or the pollution is as same as Delhi. And then I moved to Kalyani. Kalyani, it's in a remote area, very pure air. I can really say that my exacerbations or my common cold frequencies have decreased a lot.
I can believe that.
Whenever early in the morning I used to sneeze, I was in Mumbai, I used to have a mucus with some amount of grayish tinge, but I no more have this in Kalyani.
My cough goes away when I'm outside Delhi.
And that was a very nice presentation. And I think this whole concept of gut-lung axis also gained importance during the COVID times, because the ACE2 receptor, like you said, is predominantly in the lungs and in the gut.
Yes.
So, and that is the connection between the
And
gut
you
and
never
axis.
think
So
beyond
really
that. You know, we always think gut, gut, and then again lungs. Excellent,
So
excellent access.
Hippocrates has mentioned
all diseases begin in the
gut. All diseases begin in the gut. So he thinks that gut is the primary organ and most important organ, having effect on each and every system. If someone is depressed, you give him a food which he likes, his mood will get elevated. And someone who is very happy and you present him a food which he doesn't like, he'll get depressed. So we can say जैसा पियोगे पानी वैसे होएगी वाणी, जैसे खाओगे अन्न वैसा होएगा मन.
That's excellent.
Thank you.
Question? Any questions from the high?
Are you looking after cystic fibrosis as well? Because that is gut connected.
Ma'am, cystic fibrosis patients are the few things is it's rare. Second, there are challenges with diagnosis. Earlier it was earlier. ILD, yes, smokers, ILD, bronchial asthma. But only thing, when I was reviewing the literature, I got to know the importance of this prebiotics and probiotics. So I never used to upfront say give someone a prebiotic and probiotic unless they have an antibiotic-induced diarrhea or they say that my gut is having some amount of issue. But I think lung being one of the major organs and constantly exposed to air pollution as well as microbes, so we must think something about to improve this gut lung axis, and we should increase our research around this parameter, I feel.
Yeah, I mean, we didn't think beyond it, so thank you for this very
It was really
enlightening
wonderful
talk.
to hear. I'll request Anita ma'am to present a token of thanks to Dr. Jinsari. And I will request Anuja ma'am to present token of thanks to our chairpersons, Anita ma'am and Miss Anjali.
No hospital based.
Thank you so much, Anuja ma'am. Thank you, chairpersons. With this, we move on to our last but an interesting part of our symposium, and that's the panel discussion. I would like to call upon our moderator, Dr. Neha Bakshi, for the same. Dr. Neha Bakshi is, let me open. So Dr. Neha Bakshi is Assistant Professor, Department of Food and Nutrition, Lady Irwin College, University of Delhi. She is a certified diabetic educator from International Diabetes Federation. She's doctorate from Lady Irwin, MSc in Food Nutrition from Lady Irwin, and BSc Home Science also from Lady Irwin. She has a working experience. She's been part of the assistant professor since 2016. She's consultant at NCERD, Department of Food and Nutrition, from 2019 to 2020. She's a nutrition observer with Medanta Institute of Liver and Regenerative Medicine from 2012 to 2015. She's assistant nutritionist with, she was assistant nutritionist with Medanta in 2010 to 2012. She has guided 16 master theses in different areas of nutrition with more than 30 publications. She's editor and author for books. She's an expert, consultation trainer and jury at the National Olympic Association of India. And she has also given awareness through video regarding myths and facts related to dietary practices during COVID by UNICEF, SSI, and my government. With more than 13 years of experience in clinical nutrition, she has taken a keen interest in planning nutrition strategies in disease management and institution food management. We welcome you, Dr. Neha. Over to you.
Thank you, ma'am. It is a pleasure for me to invite the panelists for today's panel discussion. I would like to invite Ms. Anuja Agarwal on the dais. She is a former senior dietitian from All India Institute of Medical Science, New Delhi. She has been awarded prestigious R&D Sadhana Award 2022 India in clinical nutrition. She has received award in Excellence in Nutrition Award in 2022-2023 by International Society of Medical Food and Nutrition. She has many academic achievements with more than 34 years of experience, amazing, in the field of pediatric nutrition. She has a fellowship in pediatric nutrition from Birmingham University and a certified genetic metabolic dietitian from British Dietetic Association UK. She is certified in dietary therapies in epilepsy and autism. She is a trained dietitian for Food Map Diet in IBD and many more. So I would like to welcome Dr. Smriti Sharma here. She's a program officer at Mother, Infant, Young Child Nutrition Department. She has her work experience includes this since 2016. She is a program officer at Nutrition Tata Trust, Mumbai 2016 to 2020 and till present. She has done various projects under her, be it Comprehensive Management of Acute Malnutrition, Rajasthan, India, Project Spotlight, Palghar, Chandrapur at Maharashtra, and Assessment of Dietary Intake of Young Children, Nutrition Quality of Supplementary Food, Karnataka, India. I would like to welcome Ms. Deepti Khatuja, Registered Dietitian, Chief Clinical Nutritionist, Fortis Memorial Research Institute, Gurugram. She has about 18 plus years of experience in the field of clinical nutrition, and she has done various ASPEN LLL certification courses in be it nutrition assessment and many more. She is a life member of IDA and IAPEN. I would like to welcome Dr. Divya Chaudhary. She is the Chief Dietitian at Rajiv Gandhi Cancer Institute, New Delhi. She also has more than 18 years of experience in the healthcare sector. She is a principal nutrition advisor at the NISA Foundation NGO for cancer patients. She is PhD in nutrition from Delhi University and MSc from Delhi University. I would like to welcome Ms. Beenu Bhatia, Joint Secretary IAPEN Delhi Chapter. Ma'am is an AGM at Nutrition Research Development at Seed Fast Nutrition, and previously she has worked as an assistant professor in Department of Nutrition and Dietetics, Faculty of Applied Science, Manav Rachna, International University, Faridabad, and she has over 13 years of teaching and research experience and around 5 years of experience of clinical and industry experience. Now I would like to welcome Ms. Anjali Bhula, dietitian, All India Institute of Medical Science. She's a compassionate registered dietitian with about 20 years of experience, uh, and she has received various awards, be it certificate as Associate of the Month, January 2006 at Indraprasth Apollo Hospital, awarded the Certificate of Appreciation by Senior General Manager at Indraprasth Apollo, and many more. She has also published chapters in various books. So I welcome all the dignitaries. Actually, I prepared a small presentation and I thought that I'll just set the stage for this. And now I have to figure out where is it. Yes. Yeah. So the topic for the panel discussion is probiotic current trends and future horizon. When we talk about diet, when we talk about nutrition, as dietitians, it is actually a big challenge for all of us, because first of all we have to counsel our patient and tell them that okay, why it is important. They consider that taking a medicine and this is the end. I will be treated, but COVID did actually, it made all of us positive, but actually there was one thing positive out of COVID, that now people are talking about diet, now people are talking about nutrition, people are concerned about that how I can improve my immunity, what should I eat so that I can make it better. So today we talked about probiotics, and probiotics has great role when we talk about immunity. It has immunomodulatory properties, and when we see if we see the complete profile, we can consider that probiotic could be cost effective in the overall treatment. If we incorporate it in a diet, it can be cost effective. We will discuss this further. We discussed about probiotics. It is incomplete to talk about probiotics if we don't talk about prebiotics, which act as a food to the probiotics, and postbiotics, which are actually the metabolites. These are the metabolic byproducts of the probiotics. So various health promoting properties are there of probiotics. It is just like in the revision what we have been discussing since morning. It has a role in stimulation and regulation of immune system. Then it improves digestibility. There are in the morning, Dr. Nija shared about that how probiotics can have an impact in mental health related issues, be it depression. And when I was doing I was just searching for the literature, and I found out that it has been also been used for autism, patients with autism. Then healthy skin, so we discussed about it. It actually helps in improving the skin moisturization, and because it helps in retaining the water. And aging, we discussed about that how it can be anti-aging. And lifestyle disorders. So recently, it has been also been studied that it has an impact on diabetes and obesity. It has been shown that it has appetite suppressing impact, and it can bring about help in reducing the body weight. Then gastrointestinal disorder. So we know that it is various gastrointestinal disorder. If we consume probiotics, it has shown to be, it has shown positive effect. But yes, definitely we need more research in this area, and other chronic illnesses. We discussed about probiotics in the beginning, and actually it is the food for thought. It is something new in the conversation when we talk about probiotics. So when prebiotics are ingested, gastrointestinal bacteria ferment them, and bioactive postbiotics are the byproduct of the fermented process. So these are the different metabolites of probiotics, starting with short chain fatty acids, biosurfactants, plasmalogens, vitamins, exopolysaccharides, teichoic acid, enzymes, and bactericidins. So today in the panel discussion, we will be covering few topics, starting with cardiovascular disease, pediatrics, cancer, immunity, and probiotics. I would like to start the panel discussion by asking Ms. Beenu Bhatia. Ma'am, if you can highlight the mechanism of action of probiotics in boosting immunity.
Yeah, first of all, thank you for thank you to the organizers for inviting me for this panel discussion. As from the morning, we are talking about probiotics and we are talking about all the positive effects that probiotics has. So yes, probiotics are very important for the immunity. In a simple, like in the morning sessions, we have already covered immunity topic. So in a simple language, I will just uh, conclude this thing. First thing is probiotics prevents pathogenic bacteria from the invasion itself, as it restricts this pathogenic bacteria on the sides of the mucosal epithelial cells, thus leading to the immunal leading to the immunal more of the immunal barrier integrity. Another thing is that it also modulates the dendritic cells. It also modulates the functioning and the maturation of the dendritic cells, which again leads to the stimulation of T cells, and again it improves the immunity basically. And another process of probiotic is it stimulates cytokinins. Again, cytokinins, when cytokinins are stimulated by the probiotics, it leads to the increase in number of T cells, which again leads to the immunity process. So it, in all these things, it helps to improve the immunity of the person. It improves the immunity of the gut health, first of all. Then obviously, as we have already learned throughout the day, that gut health is related to the immunities itself.
Yeah, right. Actually, it has an impact both on innate immunity and adaptive immunity, both. I would like to ask Ms. Anjali, what is the impact of probiotics on gut microbiota, and are they healthy or they can be risky? So, just a trick, following the tricky question, that are they healthy or risky?
Okay, thank you so much for the invite. Firstly, and very much mentioned by you, Dr. Neha, like it's a revision of since morning, whatever everybody has spoken so well and so in detail. So yeah, the question, can you please repeat it?
Yeah, so what is the impact of,
yeah. So the impact of probiotics definitely is good on gut microbiota, because it helps restore the microbial balance. Firstly, it increases the immunity because it has an antibacterial effect, right? Then it certainly helps certain nutrient absorptions. And talking about the risk, actually it's healthy anyhow, but it can be of risk if the person is allergic. Like Dr. Mahesh very well mentioned it. Agar, matlab if a person says I'm allergic to curd, no, so we cannot force them to have it. So it's anyway individualized setup. So everybody reacts individually to each food. So we cannot force something on any of the individual. And secondly, certain, because we see the patients, they do say I am allergic to curd and all, and मुझे gas होता है, bloating होता है. So in that cases it can be, they definitely don't take it then.
So personalization is the
Yeah,
key,
that's the
actually.
key.
So now I would like to move to Ms. Deepti. If you can highlight the impact of probiotics and protein digestion, as we have said that it helps in digestion. So, yeah.
Thank you, Dr. Neha, and thank you, organizers. Definitely we have seen that, since morning that probiotics have a role in protein digestion. So how it actually does is there are some theories, so 4 to 5 key points. That is what has been spoken in the various theories and various researches. That first, it actually enhances and influences the intestinal microflora, which actually, results in the and have a better impact on the proteolysis, that is the protein breakdown happening in the body. Secondly, the second mechanism by which it is being researched and it is in the context is that it helps in the host enzymes increasing their peptidase activity, and also they have the ability of producing certain exoenzymes, that is outside the body, which can actually helps in the protein digestion.
The third aspect that they have that has been clearly shown is there, apart from the digestion, they are very, very much related with increased absorption by increasing the epithelium absorption to the protein breakdown. And that's where I think the plant protein, where people say that the digestibility, and in one of the talks we were actually discussing that. So that is something which is improved when you have the probiotics. The lastly and the last, not least important mechanism is that they actually inhibits the host, the bad fermentation of the proteins, and thus the toxic metabolites which are produced from the proteins breakdown and the fermentation is taken care with the help of probiotics. So these are the four different theories and the mechanisms by which it actually aids in the protein digestion and the absorption.
Yeah, it does have the proteolytic property, because of which it can help in improvement of protein digestion. Anuja ma'am, I would like to ask you that is it useful to incorporate probiotics in daily diets of children?
So, thank you so much. I've been here. So, I'm carrying this because we really don't practice probiotics in children, and Nija will not like it when I say that. It's basically because there are no recommendations. For children, if there are recommendations, we'll be free to use it. Whenever we use it, we discuss it with our team, faculty and everyone. And still the attitude, very recently, a neonatologist were discussing about probiotics, and they still said no, low evidence, right? So if people say low evidence in children, and still the studies are in the very, trial stage and all that, it's always a fear. And that's why people don't talk so freely about children on any of the forums, that they feel, for children, it has to be a recommendation for us. And there have been some recommendations from a consultative groups, which I'm going to share in the next coming questions. But at the moment, of course, they have shown in various studies, even in children, in preventing acute diarrhea, healthy gut, restoration of gut, and so many other benefits. But we still do not provide advise probiotics in children as a recommendation in any of the conditions, except for maybe diarrhea or some GI problems, and that too after consultation with the team, should we or should we not? And let me tell you, still there is an attitude of saying, though there's no harm, but there's no benefit. That is the attitude still, because there are no consultative recommendations. So we still go by the natural probiotic things in children.
So we need to get more information, more scientific evidence, especially when we talk about pediatric patients. I would like to move to Dr. Divya. Actually, when we were students at the time, we used to discuss about probiotics in a way that is just, for the preventive aspect. It is only used to boost immunity. But now it is being used in treating conditions. So I would like to know from Dr. Divya that use of probiotics in chemo prevention of cancer. What would be your comment on it?
Good afternoon, all of you. Thank you, organizers, and thank you, Dr. Nihar, for highlighting this question. Like you very well said, that we used to study the prevention aspect when we were in college. The preventive role of probiotic was boosting of immunity. This basic is actually used in the treatment when we see cancer patients. As we all know that cancer has now become almost the second largest cause of death globally, not in India. So everyone gets scared when they just hear this name. And when it's come to the treatment, now we have specific treatments where we call them as immunotherapies, where the doctors are actually targeting the immune system of the patient so that they are able to benefit better from the treatment. So there are 2-3 mechanisms where probiotics have a role, and they have been scientifically backed up. Here I would like to say first that, as somebody said, traditional wisdom. which is being prevailing since long, since being transferred from one generation to the other. But now we are living in an era where we need scientific evidence to prove everything. Earlier, there was no requirement of giving it as a proof in terms of scientific study. That is why probably it was called as an ancient wisdom. So now studies have also approved and they have established a good role how probiotics plays an important role. We all know in boosting immunity. Then they have an anti-inflammatory role. So cancer patients are struggling with a lot of inflammatory processes already in the body. So they help in that. Then they also help in apoptosis, which is the natural cell death. So every cell has a cycle. So and cancer cells, we all know that they are dividing, very great speed. So probiotics also has a role in that, and some studies have also come to a conclusion that they actually help to a patient not to reach to a metastatic stage also. So that's how definitely they have a preventive role. Role in the treatment also.
Okay. Right. So from cancer, we move to a little bit about cardiovascular. So I'd like to talk to Dr. Smriti, as you did your PhD on a role of probiotics and cardiovascular disease. So what is the mechanism of action of probiotics and prebiotics in cholesterol lowering? Is there an evidence for that?
Dr. Nihar, for asking that question, and also thank you for having me here. With regards to your question, right now the audience is pretty well aware and well versed with the advantages and the role probiotics and prebiotics play, and they have, especially in the health and well-being. If we can cover all the advantages together, amongst the most common definitely include the anti-inflammatory actions, the role on diarrhea, constipation, Crohn's disease, IBD. Cholesterol lowering and cardiovascular diseases have also been very investigated in great detail. Starting from in vitro to in vivo experiments, they have shown great impact on lowering cholesterol levels. Human studies in India, they were at the time when I did my PhD, but they were really scarce. But what to your question, coming back to a question on what's the mechanism, a disclaimer before that mechanism of action, it depends from host to host and the interaction between bacteria and host genetic structure. Given that disclaimer, it's the lactic acid bacteria and the bile salt hydrolase, which is present, especially in the lactobacillus and bifidobacterium strains, where we see the interaction with the host bile salt mechanism through which the action of cholesterol lowering is demonstrated. And how does that happen is the BSH, which is the bile salt hydrolase, deconjugates the bile salt of the host, which bile salt is actually the precursor of cholesterol is the precursor of that, and once bile salt is excreted from the gut, cholesterol makes up for the losses and hence brings down the cholesterol level in human body and thus having an impact on cholesterol on cardiovascular. Prebiotics, as now again the audience is well versed on how they improve the growth of probiotics in the gut. So one, they both work synergistically in the gut system as symbiotics when they are mixed with pre and pro, and also through their own mechanism of producing short chain fatty acids, they have an impact on lowering cholesterol.
Yeah. So, yeah, right, exactly. So there are evidences which talk about the uses of probiotics at be it cardiovascular or cancer patients. So though we are, I think every session concluded with one line that there we need more evidence. But there are actually the newer strains, newer probiotic strains are getting identified. So actually, the researchers are there are a lot of researchers which talk about it. So I would like to move to Ms. Deepti, that if you may comment on the next generation probiotic which was discovered in Northeast India. So if you can comment on that.
So this was a very recent innovation which happened, and actually from the northeastern foods that are being commonly prepared, they were able to get a strain called Lactobacillus plantarum, which is JBC5, and they have seen it was the first species that that has gone with all the recommended tests given by the ICMR to be considered as probiotic. And this is normally found in Northeast India dishes. Not, I mean, one common source that we see for most of the lactobacillus strains is the curd. But apart from that, the fermented milk and the fermented vegetables that are prepared in these northeastern states have been a very good source of this. And this strain, along with the other strain called Cinnarophytus in algans, it actually helps in the longevity. They have seen certain theories have been researched on the longevity, and basically, again, there are a lot of factors, and one of the factors is that it actually, decreases the pigmentation, the aging pigmentation, and which is called as lipofuscin, and it increases, on the other hand, the physical activity also. Apart from that, they have also seen that when you go ahead with this strain, they are still researching, and they have seen that they are able to protect aging and increase the longevity by protecting the cellular pathways from the stress and preventing it from the oxidative radical damages that are happening. And that is one of the major recent one which came out, that it can, along with fat accumulation, fat metabolism, and other aspects, it can actually it also is related with the serotonin gene protection also, and thus impacting the cognitive development. So along with longevity, they have worked on the cognitive part and also taken care on the these effects of skin and other aspects to it.
Okay. Thank you, Ms. Deepti. I would like to move to Ms. Beenu, that if you can suggest a few ways to make probiotics accessible and its usage at a day-to-day level, at a population level. We were talking about disease specific till now, that at a population level, what could be your suggestions?
See, in the morning also, in sessions also, we have discussed about this. Usage of prebiotics and probiotics, both are essential in the day-to-day life. And probiotics, as we are talking about the traditional foods in our Indian culture. So, consuming dahi, consuming buttermilk, consuming pickles, which are low in salt, and so in this form we can include more probiotics in our diet in day-to-day excess. And along with that, prebiotics, we are consuming prebiotics because we are consuming salads or we are consuming fruits and vegetables. So we are consuming prebiotics. So when we are consuming prebiotics and probiotics both hand to hand, obviously it will be giving our gut a good association of both, and it will be giving the beneficial of both the things.
Okay, so I think Ms. Deepti has already talked about that how probiotics can help in plant protein absorption. If you can add a little bit more on it.
Yeah.
So basically, what the although Dr. Neha, most of the researchers are not very clear, and in this field we really need more researchers on this. But one of the mechanism that is being thought is what they find was one of the species called Bacillus coagulant BC30, that is normally found in the curd, and is lactic acid producing, and it actually is a spore forming bacteria, and because of these lactic forming and spore forming activity, it can help in the breakdown of the protein digestion, and they can survive in our very harsh atmosphere of the stomach, which is acidic in nature. And that's, although the studies are not very clear, and normally it is thought, that the plant-based proteins are specifically later during the day and the nights, it forms a lot of bloating and the gas formation, and is not digested easable, and also the protein absorption is very low, of low value. But still, this one of the researches that showed on bacillus cannulans have actually resulted into more better researches where definitely there's an impact on the protein digestion.
Okay. Thank you, Ms. Deepti. Anuja ma'am, I would like to come to you again. And if you can, what are the therapeutic uses of probiotic in pediatric GI conditions? So, may I repeat the question, ma'am? What are the therapeutic uses of probiotic in pediatric GI conditions?
Okay, GI conditions. As I was just going through the literature, I came across there was a national consultative group which was formed by a leading gastroenterologist, Dr. Yacha, who was in SGPGI Lucknow, and there were other gastroenterologists also. It has been the recommendations is now being published in Indian Pediatrics in very recently, March 2020. So what they felt is they have come out with recommendations because, as I said, there have been studies which have shown, benefit effects in GI, especially in GI related to this thing. But they have identified a few strains like Lactobacillus GG, which is shown mild to moderate evidence, and then S. polari, which is shown the evidence is very weak in acute diarrheas, and then the mixed probiotics have shown especially advantage in NEC. Especially it has been shown very advantageous in preventing NEC to go to stage 2, preventing sepsis and even mortality. So that has shown a good evidence of mixed kind of probiotics. So there where they have given the recommendations that this could be used, and with this recommendations, because this is new to me also as I was going through the literature, so that will give a lead to pediatricians and pediatric dietitians to use it more freely. As I said, without any recommendations, using it, people get, in case, especially in case of children, people get very, very scared to use it. If, they are sometimes, people say that if too many microbes go into the gut of a child, then it may be counter beneficial also for children. So I think with this, but they have clearly said that they have no role in persistent diarrheas and something like celiac disease and other related disease in GI. They have no. Definitely, they have a role in NEC and acute.
So I would like to move to Dr. Smriti. If you may share any significant trials and their findings related to probiotics in CVD patients.
That would be an interesting one because I'll be calling my PhD thesis on this. There's no data where in trials, like I said, both on animals and humans have been done to assess the efficacy of probiotics, prebiotics, symbiotics on cholesterol lowering effects. I'll particularly take the example of the PhD data of my dissertation, wherein we had a sample of about 100 patients who were randomly distributed between the experimental arm and control group. And what we did with them, of course, the intention was to understand the efficacy in Indian subjects, number one, and also understand that what is the role probiotic if we have a symbiotic which is used in Indian subjects, what role does it play, especially when it comes to the cholesterol levels. The experimental product at play here was what is being marketed by Mother Dairy, and the strains at play were Lactobacillus LA5, Bifidobacterium B12. It also has inulin, so it was synthetic.
Control group were given traditional yogurt, and this study was done in the preventive health centers of central government health scheme, and the two centers were Janakpuri and Arkipuram. So there were a total of 100 plus samples, slightly over 100, which were randomly distributed into experimental and control groups. Of course, given it was a randomization, although we had done a series of back-end analysis on our anthropometry, which includes all height, weight, BMI, waist circumference, hip circumference, your body fat percentage, from anthropometry to nutritional, dietary call, to clinical, as well as, of course, like blood pressures, to blood draw, which included, that's where we will come to know the cholesterol, the lipid profile of the participant. Before running the analysis, we did check for the comparison, and all these parameters were comparable between the experimental and control group. And of course, where we did not find comparison was waist circumference, for which the data was adjusted accordingly. We found a significant improvement towards endline, which was an 8-week intervention, and each participant was given 400 grams of either the symbiotic yogurt or the traditional yogurt. At the end of 8 weeks duration, and the lab tests were again done by the NABL accredited labs, we found significant reduction in the lipid profile. We saw a downward trend in in blood pressure, especially the systolic. We ran both intention to treat analysis as well as per protocol analysis as per the recent guidelines. We saw about 7 to 8% in total cholesterol and LDL cholesterol reduction, about 12 to 14% reduction in triglycerides and VLDL cholesterol respectively. So broadly, these were our findings, and there was an increase in dietary fiber thanks to the inulin which was there in the yogurt. So overall, our patients towards the end of the trial, when we closed on the findings, they were educated on the use of probiotics. So here, the key was that it was compared with the traditional yogurt, which Indians usually consume.
Okay, thank you, Dr. Smriti. I would like to move to Dr. Divya, if you can tell us about the mechanism of probiotic action in cancer prevention and therapy.
We've already covered the mechanism of action for prevention in the earlier question. So I think we'll talk about therapy. Probiotics definitely have important role, specifically if I talk about cases of cancer of colon, rectum, or basically GI issues. And I would like to mention here that I did a small study in about 50 subjects in my hospital. And they were regularly given probiotics for a specific period of time. And we found that there were considerable improvement in the GI symptoms of the patients. There was a reduction in flatulence, there was a reduction in the persistence of diarrhea, and the bloating also was very much reduced. And because of improvement in these symptoms, the oral intake of the patient was getting better on a daily basis. Because if they were regularly complaining of diarrhea or bloating or flatulence, they were not eating well. And eating a balanced diet for a cancer patient in itself is very challenging. And over a period of time, if they're not eating well, they will be losing weight, and other side effects will crop up. So it definitely has a very beneficial and very important role, specifically I would say in GI cancers.
And also, if you can talk about the probiotics influence on other mutagenic and carcinogenic factors.
Yes, definitely. What happens is basically because of radiation enteritis or because of side effects of chemotherapy being given to a patient, there are a lot of GI disturbances. People are burdened with either having diarrhea most of the time. If not, so they will be constipated. And we all know that chemotherapy produces lots of toxic metabolites which have to be regularly removed from our system. So probiotics, in addition to giving relief from these symptoms, help to remove these toxic metabolites from our bodies, so that the pathway for absorption of nutrients is clear, and there are chances of very less side effects, and that's how it helps to eliminate those toxins from our system.
Okay. Ma'am, I would like to move to Anjali, ma'am, for the role of probiotics in chronic GI disorders like pancreatitis, gastric ulcers, and even their role in IBD and IBS. You can highlight.
See, as we all know, pancreatitis is definitely inflammation of the pancreas, and gastric ulcers are, open sores in the lining of the stomach, right? So definitely probiotics, as of my personal experience also, they definitely work well with these cases. They, the patients get relief from the symptoms. Like with pancreatitis, definitely abdominal pain is associated. So it gives relief in the pain. It gives a cooling and soothing effect, right? The patients definitely they come up with the good results having probiotics, curd, lassi, whatever you say, right? And saying for IBD and IBS, this also definitely the stool frequency it reduces, the consistency of the stool reduces. Anyway, any of the gastric issues, probiotics for sure works very nicely. The patients with personal experience, like Dr. Um, Saurabh Kedia had mentioned, we don't have any recommendations, but clinical trials do prove the beneficial effects of probiotics on GI symptoms, gastro patients. The recommendations and studies definitely we should be on to do it.
I would like to add just a small thing to what Anjali said, that definitely I have also seen in my patients there is a reduction in the frequency of stool passage, specifically where patients have been operated for colon surgery
Yes.
and they have a colostomy bag. They are basically running to the washroom after every hour or every 2 hours. So by supplementing their diet with probiotics, I have seen personally definitely there's a reduction in the passage, in the frequency of passages.
Yeah, even I'd like to add, like I see patients of GI surgery like ileostomy, colostomy, they अगर हम if they're not properly counseled, right? So अब कितनी बार बैग खाली करें तो they'll the whole day they're doing the same thing, 5 बार 6 बार, and after we counsel them so nicely, because we have patients of all economic status, right? So ऐसा नहीं कि हम बोलेंगे आप मतलब खरीद लो ये तो they'll not. So we go on curd and all. We restrict them on milk and more on probiotic supplements. So they do well on that. The frequency definitely reduces. And the consistency too.
This also gives a secondary advantage in terms of if they are cleaning back quite often, they have
Yes.
to open it. So it opens the door to more pathogenic bacteria because it's a wound. So then there are chances for the patient to catch secondary infections along with that. So that becomes a host of issues for the patient. So directly, indirectly, probiotics have been shown to give them very beneficial.
So basically, I think that the conclusion of the thing should be जो दादी नानी कहती थी ना, that's the घर का खाना, not घर पे खाना. Right? There's a major difference between the two.
Since it's a discussion, if there are any comments or any question in between, we can take it. So if any delegate would like to ask or share comments, can please.
About I just wanted
Yeah,
to add a
yeah,
comment
please.
that about the cholesterol reduction. Of course, you spoke about the mechanism, but also there are a lot of studies which have shown that the probiotic bacteria can actually bind to cholesterol and remove it from the system. And also for carcinogens and mutagens, there are studies which show that probiotics have the ability to bind to them and thereby remove them from the.
And since they have an anti-inflammatory response also, so majority of the patients, whether from oncology background or GI disorders or cardiology, all of them are struggling with inflammation. So we are loading them with steroids and other medication to reduce. Even if we give them a daily supplementation of probiotic in the form of food, so that definitely helps. It reduces, कभी-कभी patient बोलते हैं सिर्फ दवाइयाँ ही खाएं क्या खाना कब खाएं? It's like that.
So I'd like to move to Binu ma'am. Ma'am, if you can just tell that general public that what is the what should be the recommended dose if they want to, consume probiotics?
As such, there is no recommended dose as from the morning we are talking about. But yes, in a normal like day-to-day life also, through the review papers, it has been seen that 5 to 10 billion CFU is sufficient if a person consumes on a daily basis. That is, it's not a recommendation given by anybody, but on a review papers, it has been seen. And as FSSAI has also declared, like Dr. Bhavna has already stated about that. So 10 raised to the 8 power of CFU, they have told, they have recommended to be added wherever they are adding in the probiotics, or if they are adding in any of the supplements which include probiotics along with the prebiotics, then the strain also, the strain name also should be mentioned, and the quantity of the strain should also be mentioned. And as such, the recommended doses, there is no such doses regarding the probiotic has been given. And for the prebiotic also, there is no such recommendation has been given. But yes, for the prebiotic, we say that the fiber intake they have given like 40 gram on 2000 kilocalorie of diet.
Yeah.
So for the prebiotic, still we can say that okay, 40 gram per day you can have on a 2000 kilocalorie of diet. But for probiotics, there is no recommendation as such. But if we are depending on the evidence, then we can suggest that 5 to 10 billion CFU can be taken on a daily basis in a normal person.
Also, ma'am, to add to it, if you can talk about its cost effectiveness, the probiotics and prebiotics
So
at a
if-
general level.
If we are talking about the probiotics cost effectiveness, one thing is like the traditional method which we are using, the home prepared things we can include in our diet. That is automatically cost effective. We are included in a balanced diet. Another thing, if we go for the supplements, or if we go for the probiotics which are available in the market, in that case, there are few points which should be kept in the mind while selecting those probiotics. First thing is the strain, which kind of strain it is having. Secondly, the quantity of the strain. Thirdly, how it will be stored, because yes, the storage part is very important, because live microorganisms, they do not stay for a longer time. If the storage like temperature is not proper, and the storage capacity is not proper, so they can't stay for a longer time. So it's of no benefit if you are consuming after, because normally अगर इंडियन उसमें भी हम देखते हैं बहुत लोग जैसे याकुल्ट भी है, people are getting it from the market but उसको बाहर रख देते हैं. So it's of no use. आप उसको 3 घंटे बाद 4 घंटे के बाद अगर आप उसे कंज्यूम कर रहे हैं तो उसका बेनिफिशियल इफेक्ट जो है वो नहीं होता. So that is very important. People should be aware of all those things. Another thing is expiration date. Expiry is very important. अगर expired product है and if you are consuming it, कि कोई बात नहीं इसका, because there are misconceptions in the mind कि potency कम हो जाएगी. ऐसा नहीं होता है. But अगर expired है तो probiotics के case में it is of no beneficial effect. And another thing is whatever the probiotics we are choosing, it should help in our gut health. Like it should be it should be more colonizing in our gut. Then only it will give us the beneficial effect. So these points should be kept in the mind when we are choosing the probiotics from the market. And other than that, traditional, like with as I have already told, that buttermilk, dahi, kanji, which is used in
Food
Indian
sources.
house. Yeah.
we can-
So food sources which we can include. That is the most cost effective way.
If I can just add to you,
Yeah.
Devi. So you were absolutely right. So if you see, FSSAI has now brought out standards for probiotics. They say that any product which is calls itself a probiotic has to have 10 to the power 8 CFU per daily serving. So any product which contains 10 to the power 8 CFU. But what is very, very important is also, like you said, the strain of the bacteria. So every product is not going to work at a particular level.
Yeah.
So there will be changes. and there
That's
will be
why I added that the strain quality, name of the strain should be mentioned, and it is important to choose which kind of strain. As from the from the daily from today's talk, we have concluded, because no one strain is recommended
you
for.
the same. And also when we're talking about a lot about traditional fermented foods, but then in the market we have curd and we also have probiotic curd. So the difference is very, very important because the probiotic curd contains that probiotic bacteria.
bacteria.
So while kanji and all the traditional fermented foods are excellent for nutrition, they do not classify as
Yes, the
true
quantity.
probiotics.
Yes, in that
No, but the
quantity
normal
is not
probiotics are not
Bacteria are
affordable.
filled with the stomach.
Yeah, but the probiotics like Yakult, if I say my patients in AIMS
Yeah,
Yakult,
that goes
so
up now.
affordable.
That is another
Haan.
story. But if
So
you're
what do you
wanting-
comment about that golgappe ka pani? Because see, if you're drinking kanji, that's homemade. So how do we comment on which
Yeah,
is
so I really
nobody's
want-
going to leave it, I think.
No, I really wanted to put this out there because I think there is this confusion that when we are talking about probiotics, for instance, we are talking about a scientifically
Yeah,
tested
definitely.
bacteria. So these products, they are excellent. They're a part of our diet. I mean, I cannot have my lunch without that katori of dahi. I will have it. But then I am not looking for a probiotic benefit in that.
Definitely.
I am looking for a probiotic benefit, not in, I'm not talking about Yapa, any product which has that scientifically validated probiotic bacteria is a probiotic. So
That-
there is this, continues to be this, and that is why probiotics is another category altogether.
That is why we have in definition adequate amount of the live microorganism as per FAO. This is the definition.
And give you a scientifically proven.
No, but I think Binu was also right, ma'am, on that way. Like if I am buying a Yakult from you, अब आपके वहाँ से delivery हुई right temperature पर, अब मैंने रख दिया, अब 10 मिनट आधा घंटा बाहर रहा, तो it's I think all gone.
10 minute आधे घंटे में, see, I'll tell you again, what is the whole thing about the expiry is that when you're looking at Yakult, for instance, it has this probiotic bacteria which starts multiplying at 37 degrees. So you have refrigerated it. It is sleeping, like we heard
Yeah,
from Dr. Vijay. It's metabolically inactive. The minute you bring it out, the temperature has to touch 37 degrees for it to again start multiplying, and then the pH has to drop for the numbers to drop. So that आधा पौना एक घंटा में
कुछ नहीं होगा.
But yeah, if you're keeping it out for 3 days, 4 days, then definitely it is not a count.
तो मतलब हम जाते हैं और पी लें.
जैसे दही को बाहर रखते हैं.
हाँ.
If we keep it out for more than 10 hours, it will become katta.
हाँ.
Simply because they're fermenting and producing more lactic acid.
I just joined on one of my colleague, I'll not name her obviously. She had a stomach upset. उसने मुझे हमें आके बताया कल मेरा पेट बहुत खराब था लेकिन मैंने गोलगप्पे वाला देखा, so I couldn't leave it, and she was okay with it.
Yeah.
So Indian stomach I think is immune to all.
Indian traditional food is a must.
Indian stomach is immune to all.
That's the over there because she was asking about the cost effectiveness.
Yeah.
So I
Yeah.
was talking about both the things
Yeah.
because see, those people who cannot afford,
Yeah.
if they are suffering from something or the other, then also they will afford the supplements which are on a lower price.
Like dahi and whatever.
So over there, I was talking about both the things. So
But
for
this
the
only thing,
probiotics,
this thing is because that whole concept of probiotics is again that fibers. Prebiotics are fibers, but
Yes.
all fibers are not prebiotics.
Yeah.
No.
Yeah,
So
right.
it has to be scientifically validated, and that 40 gram per 2000 kilocalorie is the scientifically validated
Yes.
prebiotic. Not just any fiber ले लिया उसका 40 gram ले लिया, that doesn't become a prebiotic. That
So-
little clarity is just what I was trying to bring out here.
So our discussion became more yummy with golgappe ka pani. So I would like to conclude here this panel discussion by saying that yes, there are health benefits.
No, sir,
Yeah, sir, question. Yeah.
No, these questions are not from me, but the YouTube viewers.
Okay.
I'm happy to inform you that there are more than 660 viewers on YouTube now.
yeah.
So there are lots of questions. I have just most of them have been answered. Someone asked, what is the example of symbiotic?
Probiotic and prebiotic.
Example, practical. If you want to give symbiotic, what would you give?
Onion raita or something like that. Onion raita.
But not a true symbiotic, but
Yeah.
But we have prebiotic separately, probiotic, and is there any combination available in terms of
Curd, banana and curd. Banana is a prebiotic.
He's right.
Banana and curd. I mean, this has been the traditional practice of feeding a child. That banana with little bit of curd,
Aloo raita.
potato and curd. So there we
So you mix up
Yeah.
and then prepare it.
Yeah,
huh?
because it's fiber and usually the source is when we talk about
Yeah,
curd
definitely.
rice, yes.
Adding
Yes.
flax seeds also.
So discussion is becoming more tasty.
It's a little bit a clinical question. Sorry.
Should be probiotic curd. If you're mixing onion to it for inulin, then you are actually mixing pro by definition.
Yeah, probiotic by
and by
definition. Yeah.
But it has just with that.
So basically, what we are saying is that don't go by wherever you see there is a fiber, take it, or there is a prebiotic, take it. You must know exactly which is scientifically proven.
Yes.
All of them are not exactly what they show or what we read. That is. Now, someone is asking this question whether if anyone can answer. I can't. The role of prebiotic in autoimmune disease, particularly lupus. Well, autoimmune disease may not be very specific. So I think we don't have any clinician to answer this, and this is a medical question. So I don't think.
Lupus, I don't know, sir, but autoimmune diseases like in children, autism, ADHD, ataxia has shown a beneficial this thing. I didn't talk about it because the question was not related to neurological disorders, but lupus, particularly because it's very rare, it's not being seen. Yeah.
Anything pertaining to allergy?
Allergy, yes, yes.
So
Yeah, there is a lot of scientific evidence which talk about that probiotics can be it is
and Dr. Mahesh
anti-allergic.
also, I think, said about that regarding the role of this in allergies. And should they be taken in empty stomach or after food?
They can be
Probiotics are taken anytime during the day.
Any
They
time.
can be taken, but normally you have it after a meal simply because your acidity levels are also lower. So it's just allows for better survival of the
But like
probiotic,
we
but
take-
anytime during the meal.
But like we take rice kanji, if we take an empty stomach, it's very much beneficial. We soak cooked rice in a water overnight, and then we take it with a slight turn of a curry leaf.
Are they talking about the
Like
probiotic medicine or they're talking about drug or they're talking about probiotic food?
You have to take in the morning. Maximum they say it should be taken early morning, but it is strain specific.
Yeah.
Like lactobacillus cerota can be taken any time of a day, and generally it is being administered post lunch or so. But generally, on an average, we can say that it should be taken empty stomach in the morning.
Yeah, like if you say homemade product like dahi, जब बच्चा जाता है फिर दही चीनी खा ले हाँ ला देंगे कटोरी भर के कौन रखेगा बाद में
Contraindications of probiotics? Any?
Yeah, in critically ill, immunocompromised, it's
Yeah.
widely administered with care, but again scientifically.
Scientifically. So I would like to conclude this interesting panel discussion that health benefits of probiotics have been studied over a period of time. Its use in preventive aspect, we are very well aware about it. But when we talk about curative aspect or when we talk about vulnerable group of patients like pediatric patients, like ma'am said, we need more scientific evidence to prove it. But we cannot deny from the fact that yes, they do have a lot of positive health benefits, and we need if we apply it, we need to follow a team approach. We need to ensure that it should be undertaken in medical supervision when we are putting into practice for patients specifically. Thank you so much. Thank you.
So I will say that if anybody interested in pediatrics, I think should go to the recommendation which says conditional usage of probiotics in children, but it is a good read in Indian Pediatrics, March 2022.
And also the ESFAN guidelines, the European Society.
Yeah,
Hmm.
European Society is also given recommendations. Yes.
Thank you. Thank you. Thank you all the panelists. Thank you. Thank you. Nice to meet you, Nihar. When I was intern at AIIMS, we were the junior dietitian.